Okay, good morning, everybody. My name is Adam Kubas. I'm the director of the Institute of Physical Chemistry of the Polish Academy of Sciences. It's my pleasure to welcome you today. I must say that today and tomorrow are the days to be here at the Institute. Today and tomorrow, the best science will happen here. There will be a festival of science happening at the Institute of Physical Chemistry. It's already 11 years that we are co-organizing the Dream Chemistry Award competition. It brings to the Institute and to our partner institution in Prague, the Institute of Organic Chemistry and Biochemistry, the brightest, youngest scientists all around the world. First, I would like to welcome our finalists, 5 finalists that will deliver the talks today. I welcome the judges that graded the projects. You've done a wonderful job. And I welcome you here. As I said, this is the Festival of Science. Enjoy it. Take as much as possible out of it, because this will be the chemistry of tomorrow, as always Robert says. And now I would like to welcome and give the stage to Paweł Julgwind from Prague, also to say a few words from his side.
Thank you very much, Adam, and thank you for having me here. It's a great pleasure to do this together with Robert and the Polish Academy of Sciences. So when I was a teenager, I was a competitive ski runner, and before the real competitors started, there were always the guys who smoothed the pre-runners, right? Who smoothed the slope. So I'm gonna smooth the slope a little bit for you, and then pass it to Robert. And what I want to say is, I'm so happy that we're doing this together in the old tradition of Poland and Czechia being one country. So we are one scientific country in this in chemistry, and of course this was Robert's idea. More than now 10 years ago, 12 years ago, and we kind of jumped on the train a bit later, and I'm very happy we're doing this together. So this year it's in Warsaw, next year it will be in Prague, and then it will be again alternate between Warsaw and Prague. And yeah, maybe a little bit about what's the basic idea of this competition. I mean, here in Europe we all talk about ERC grants, right? So this is not about ERC grants. ERC grants are for 5 years, right? This is like dreaming beyond ERC, 10 years, 20 years, right? That's the kind of size of the dream. But at the same time, the dreams should be anchored, right? So in this sense, it should be a little bit like ERC, so that in 10 or 20 years, there is at least like a 1% chance that this will actually happen. So that's like, that's how we chose you finalists from the pool of applicants. And that's how we kind of, will view or judge, if you wish. No, we'll celebrate the talks that you will be giving. So we're looking for dreams that are beyond ERC, but still possible, at least in principle. And that's all I want to say, and then I'm gonna enjoy it. And Robert, stage is yours.
Thank you, Paweł. I'm gonna enjoy it as you do. You see, the scientific dreams of today is the hard science of tomorrow. So what you will hear today, you will witness in maybe 20 years as a modern science, right? And what is special about this award is that we all dream. But this is a single award for dreaming. There are a lot of awards for dancing, singing, skiing, but not for dreaming, except for the Dream Chemistry Award. And today, as it was said, you will see a Festival of Science. They are 5 excellent researchers who are all the winners of this competition. So it is not a competition because they are already winners. And you will hear the visionary science, the dreams. I'm really anxious to see it, so I will stop at this point and give a floor to Raya, our first speaker. Thank you.
It's very exciting to be here. Thank you everyone for coming. I'm Raya Sorkin from Tel Aviv University. I started my lab at the School of Chemistry 4 years ago, and I'm very happy to have this opportunity today to share my dream with you. I want to thank Robert and Pavel for inviting me to submit my application, and to the committee for choosing me to be among the finalists. It's really a great honor. So let's start, and the dream is about developing novel non-hormonal contraceptives. The lives of each and every one of us has started by a single membrane fusion event, which occurred between the membrane of the sperm and the membrane of the egg. Membrane fusion entails mixing of two lipid bilayers, so the lipids themselves merge, the membranes themselves merge, and that allows mixing of the content that the membranes enclose. My dream is to understand the basic mechanism of egg-sperm fusion, and by understanding this mechanism, to develop a new method to inhibit this process effectively towards developing new contraceptive methods. New contraceptives are really critically needed. There are 120 million unintended pregnancies per year, which account for 50% of all the pregnancies across the globe. Currently available contraceptive methods have their limitations and problems, and are not suitable for a lot of people. They are associated with side effects, so there is really a need for new methods. My dream project starts from characterizing the interactions that are at the heart of this fusion reaction. There are two proteins I will tell you more about, one on the membrane of the egg and one on the membrane of the sperm, which need to interact in order to facilitate this reaction. I then want to screen for small molecule inhibitors, test these inhibitors, and then perform the standard ways of drug discovery, of toxicity, dose optimization, and finally perform IVF measurements and in vivo evaluation of the selected inhibitors. So basically this project really goes from the basic understanding of the interaction by using force spectroscopy and developing new fusion assays, through screening of small molecule libraries, high throughput screening, and then in vitro toxicity testing, activity validation, and finally in vivo application in mice to test the effectivity of the molecules. So the first step of this project is to understand membrane fusion in fertilization. And what do I mean by that? So first I need to tell you in a few words about membrane fusion in general. So membrane fusion occurs in a lot of physiological processes. It occurs all the time in our bodies, and it can happen across different time scales and length scales. The nerve cells in our brain their activity is mediated by fusion of synaptic vesicles that contain neurotransmitter with the presynaptic membrane, which allows release of the content. So this happens very, very fast. So it's faster than milliseconds, and these vesicles are very small. They are 40 nanometers in size. Another fusion process that occurs in our body is that of myoblast fusion. So the process that is responsible for formation of muscle fibers in our body starts from fusion between cells called myoblasts, which form syncytia, which is a large cell that has a lot of nuclei inside of it. And this happens on a scale of tens of microns, so cells are on the order of 10 micron size, and this takes hours. Another membrane fusion event that we're all familiar with after the corona pandemic, so a lot of pathogenic viruses like the corona, they have a membrane envelope, and in order to infect the target cell, their membrane needs to fuse with that of the target cell to facilitate the infection. So these are just a few examples for very different membrane fusion events. And while they can happen on different time scales and length scales, all of these events are believed to have similar intermediate steps, and that is because essentially we have two membranes that need to come together, they need to attach to each other, and then there is the formation of the contact, there is formation of a stalk, and expansion of the stalk, and finally formation of the fusion pore. So this process has to be tightly regulated. It's not a spontaneous process, and there are energy barriers associated with the different intermediate steps. And that's a good thing, right? Because we have a lot of cells in our body, and they don't just spontaneously fuse with each other. Otherwise, we would have been unicellular organisms, and we are not. So fusion has to be tightly regulated. And usually, there are specialized proteins which are needed in order to overcome these energy barriers. So a lot is known about fusion proteins in nerve cell fusion and in viral fusion, but far less is known about fusion between egg and sperm, which is the focus of my DREAM project. Fusion between egg and sperm occurs at a very particular part of the egg, which is covered by microvilli structures, which are membrane structures that have a very small cross-section. And there are several proteins that have been discovered to be essential for fusion in fertilization, but it's not really known or clear yet what is the role of these proteins. So this whole field is quite new. For example, the Juno protein has been discovered 10 years ago, and all of the other proteins, a lot of the other proteins here are even more recent. And for example, Izumo and Juno, if you knock out these proteins in a mouse, the mouse will be sterile. So they are critical for the fusion reaction, and it's not really clear what is their exact role. So it could be mediating adhesion, which precedes fusion, and there is a recent study suggesting that Izumo might be actually a fusogen, though not a very efficient one by itself. So the focus of this DREAM project is on this protein pair, Izumo on the membrane of the sperm and Juno on the membrane of the egg, two proteins which need to interact in order for the fusion reaction to take place. And this is really the only known protein pair which is necessary for fusion. So in order to understand the interaction between these proteins and their roles, I want to start by characterizing the interaction forces associated with their binding. And in order to do that, I want to use natural membranes with embedded proteins. The starting point of this experiment is having cell lines which are stably transfected with the proteins of interest, one cell line for Izumo and one for Juno, and we can then induce formation of vesicles from these cells. We can collect these vesicles and we can use them to coat microspheres, to coat small beads. We usually use 3-micron beads. They're made of polystyrene or silica. We can coat them with a natural membrane, and we can use these beads as handles in order to measure interactions between the two proteins. And the really nice thing about using this method is that it allows us to obtain our proteins of interest in a membrane environment. These are transmembrane proteins, and they would have a natural lipid environment, and we will have the correct orientation. So that's a nice way of avoiding reconstitution of proteins. And then after we have these handles, we have these beads that are coated with a membrane, we can then hold two such beads in two optical traps, and we can bring them together and separate them and measure the forces that are needed to separate the two beads. Yeah, thanks. Okay, so you can see in this movie an example of such an experiment, there are two beads. They are held in two optical traps. They are brought into contact and separated, and then you see these peaks here of the forces that are needed to pull them apart. Okay, so here you see a force versus time trace, and we can quantify the forces and also the probability of interactions between the proteins. And you can see that the probability of forming interactions between Izumo and Juno is much higher than between Izumo versus Izumo or Juno versus Juno. So this is a preliminary data from a lab, and it's a starting point for this DREAM project. But understanding the interaction between the two proteins is just the first step. Basically, I want to follow this quote by Richard Feynman, who famously said, "What I can't create, I don't understand." So I really want to understand the fusion reaction in fertilization by finding the minimal system which will have fusion. So I want to understand what is the minimal set of components, which lipids and which proteins are needed in order to be able to observe fusion in a simplified system, and that will help us to understand fusion in fertilization. How can we find a minimal system that exhibits fusion? We can go either bottom up or top down. So we can either make synthetic membranes and add some proteins, or we can take gametes and take out some components. And the first step for this is the following essay. So very recently came out a very interesting paper that showed that if you take cells, just a cell line, HEK cells, whichever cells you have in your lab, and you transfect them with Juno, so these are just somatic cells expressing Juno, and here it's the mouse isoform of Juno. Then if you add some mouse sperm to your plate, the cells will start fusing with each other. So the sperm is inducing fusion of the cells, and we were able to recreate this assay in my lab. So this is an example. So here you see this cell colored in magenta and another one colored in green. These are cells that express Juno. The Juno is not labeled, and the cytosol is labeled by two different colors. So these are the same cells, and we label them in order to be able to observe fusion. And then when we mix these cells with sperm, we observe formation of syncytia. As you see here, this is a big cell with 4 nuclei inside. And here is another example. So the green contour here is a large cell. It has 2 or maybe 3 nuclei here. And you see these very small blue things here are the nuclei of the sperm which fused and entered into the cell. So this is data from my lab, but following a recently published essay. So the next step would be to further simplify the system, going from top down, and to understand what is the minimal set of components. But it might be the case that these components are not only proteins and lipids. So it might be the case that proteins and lipids are not enough in order to have fusion in fertilization, and other factors, biophysical factors and geometrical factors of the membranes might be very important as well. And in particular, membrane curvature is a very important factor, most likely, and that is because, as I mentioned, the fusion between egg and sperm occurs at this part of the egg, which is covered by microvilli. So this is an EM image of microvilli. These are tubes made of membrane. They are 50 nanometers in cross-section. And these microvilli are enriched with a protein called CD9. And this protein, if you knock out this protein in a mouse, the mouse will be sterile. And not only will it be sterile, it will also not have this morphology on the eggs. It will not have any microvilli. And that led me to suspect that this protein might be a curvature sensor and a curvature inducer, perhaps. And in order to test that, we wanted to use the combination of micropipette aspiration with optical tweezers and confocal fluorescence. So the assay is the following. Here you see a giant unilamellar vesicle, which is being held by a micropipette. There is a micropipette here. And then there is an optically trapped bead, which is pushed into the vesicle and then pulled away to form a tube. And we can control the diameter of this tube by comparing the aspiration, by controlling the aspiration pressure in the pipette. So if you have low aspiration, here you see there is a very small tongue, we call this part of the vesicle. And if you aspirate more strongly, it grows. And then you will have a more narrow tube forming. And by using this assay, we discovered that indeed this protein CD9, the egg protein, is a curvature sensor. So we view this as basically a biomimetic system because we have this very large vesicle and very small tube, and that is analogous to the large egg cell and the microvilli. And what we see is that when we increase the curvature, when we decrease the size of the tube, we have more of the protein migrating into the tube. So here there are two labels, two colors. The red color is a general lipophilic dye embedded in the membrane, and the green color is a GFP molecule which is attached to our protein, to the CD9. And you see that at low tension, the tube has both red and green color, but when you increase the tension, meaning that you shrink the tube radius, you see that it becomes more green, meaning that the protein is partitioning preferentially into this high curvature. And that helps us to understand why this protein is localized on the microvilli and probably also contributing to their formation. So in our search for the minimal system that exhibits fusion in fertilization, we should consider membranes proteins and also additional factors like the curvature in order to find this holy grail of the minimal fusion exhibiting system. So after this step, we want to move on to screen for small molecule inhibitors, because what we want to do is to understand this interaction between Izumo and Juno, understand what is the minimal system, and then we want to screen through libraries of molecules and find molecules that can interfere with this interaction between Izumo and Juno proteins. And one way we can do this is by using standard approaches in drug discovery. So we can do high throughput screening where, for example, we can use the following assay. So this is preliminary data where we expressed the soluble fragments of Izumo and Juno. We immobilized the Juno on the plate surface. So this was using his tag and anti-his plate. There are different ways to do it. And then we can introduce soluble Izumo, and we can validate whether the Izumo is binding the Juno. There are different ways. You can use the fluorescently labeled antibodies or enzyme conjugated antibodies that would have some a color reaction that can be measured in a plate reader. And this is preliminary data that's showing that indeed we can detect a specific binding between Juno and Izumo, and this is a starting point for performing large-scale, high-throughput screening where we would add different kinds of molecules and see which of the molecules interfere with this binding between Izumo and Juno. There are also other approaches for screening molecules. There are available DNA barcoded libraries which we can use for screening in solution. So that's another approach that we will consider. After screening and finding our initial hits, we will need to perform additional steps. We will need to do chemical expansion, optimization, and select our lead and backup chemical scaffolds. So we will start by evaluating the dose response of our molecules. And that will help us to filter out false positives. We can do that, for example, using isothermal titration calorimetry, which would measure the difference in the heat change as a result of binding between the molecule and the protein. So these molecules would either bind to Izumo or to Juno, and we would expect to have a dose-dependent response of our active molecules. We will then perform chemical expansion to create an extended set of analogs, and then perform structure-activity relationship analysis, and that will allow us to identify which parts of the molecules are critical for their function and find the most active chemical scaffolds. After filtering, using this to filter the most promising lead molecules, we will take a small set of molecules for a more thorough analysis and investigation, and we want to perform more detailed tests of the most promising candidates, of the most promising small molecules. And we will then go back to the force spectroscopy assay I mentioned in the beginning. And we use this approach for a different problem, for the question of coronavirus fusion and interactions between spike and ACE. So we demonstrated that you can use this way of taking natural membranes, putting them on beads, and then measuring the interactions. And you can also measure very nicely the blocking of the interaction when you add a certain molecule that interferes with this binding. So you can see here the probability of interactions of ACE versus spike, ACE versus ACE, spike versus spike. And here this is ACE versus spike in the presence of an antibody which blocks the interaction and an antibody which binds the spike. And you see that it successfully blocks the interaction because the probability is decreased and the forces are also decreased. If you look at this purple plot compared to the orange one. So we will also use this kind of assay for in our Izumo-Juno interaction assay to test whether indeed the molecules that we find interfere with this specific interaction between Izumo and Juno. And of course, the next step will be to test whether the molecules are able to inhibit fusion in a simplified assay. The nice thing about finding a simplified assay is that you would need to use much less animal material. So hopefully that works. And finally, we will do toxicity, activity, and efficiency testing. We will need to add these molecules to the gametes, see that they don't interfere with the viability, evaluate the inhibition efficiency using IVF, test whether these molecules actually bind to the gametes, and perform in vivo evaluation of our selected molecules as contraceptives. So basically evaluate the fertility rate after mating following the administration to mice. And of course, in the final step, we will need also to consider the administration route and think whether this will be a pill or a patch, and maybe we can think of targeted delivery of these molecules for effective activity. So this project really goes from the basic understanding of the interactions between proteins that are at the heart of this reaction, developing assays, screening, testing, optimization, and finally in vivo tests. It's important to note that there will be a few iterative steps here because when we change the chemistry of the molecule, it might affect also the toxicity and the activity. So there will be a lot of back and forth in these steps of screening, testing, and optimization. And hopefully in the end, we will find our promising molecules that can be candidates for new contraceptives. When you have big dreams, it's really good to find people that can share your dreams and help you realize them. So I'm very happy to have found amazing collaborators that join me in the pursuit of realizing this dream. So I've teamed up with Dr. Maayan Gal. He's an expert in protein interactions and drug discovery. Professor Ruth Chalgi, she's a reproductive physiology expert, and Dr. Jerome Tubiana, a machine learning expert for structural biology. So hopefully together we can realize this dream. And of course, together also with my amazing group. So this is my group. I'm very grateful that those people chose to work with me. I also want to acknowledge our funding, and thank you very much for your attention.
Thank you, Raya. Are there any questions? Directors, the first one.
Yeah, thank you very much. Wonderful dream. Short question. So, if you have a contraceptive, you would like to have it reversible contraceptive.
Yes.
So, and you will spend a lot of time of making molecule that will attach to a protein, but then you have to have a way to take the molecule out of it. So, can you say us a few words how you would like to approach this problem?
Yeah, that's a very good point. It's also the question of whether this will be a male contraceptive or a female contraceptive. So if it's a female contraceptive and it's binding to the egg, so then there is only one egg that is ready, right? So that will be fine, even if it's not reversible. But of course, we need to make sure these molecules don't cause any damage. And also, if it's going to be for a sperm, then will be the question of crossing the blood-testis barrier, which is another issue. And the sperm is regenerated every 3 months. So yeah, maybe it's fine even if it's not reversible.
You just stop the tape.
Yeah, that's the idea.
Okay, next question.
I have actually a couple questions, but hopefully short. So the first is the way you select the target molecules as inhibitors. So if I get it right, you will do essentially a blind screening of libraries. So
Yes, that's the idea.
is there a way how to do it maybe more efficiently than just screening whatever's there in the library? And are you thinking about that?
Yeah, that's a very good point. We have a certain direction together with the machine learning guy, so he had some ideas how to do that. Yeah, I prefer not to say too much about that, but indeed we are thinking about more rational design of molecules, which will probably be more effective.
And just a second question, if you could target it directly to, say, the egg or the sperms, you could maybe play with lowering the dose. So are you thinking about how to specifically, have
targeted delivery
where it should be, and so it doesn't poison other cells in a sense?
Yeah, that's a very good point as well. We were thinking about this a lot. We don't have a solution yet, but for sure it will be best to have targeted delivery, which maybe will be targeted to a molecule which is specific for the sperm or the egg. There are certain protein candidates which can play that role. So ideally, yes, we would think of a targeted delivery that would be targeted to a specific protein. Yeah.
Thank you.
Okay, so your approach is based on the long exposure of this molecule to the receptor. And there are many cases actually that if you have the long exposure, you can have the mutation of the receptor and then your molecule will not work. So how would you comment on this?
Let me see if I understand. You're saying that the protein itself, the Izumo or the Juno, would undergo mutations in the presence of the molecule.
Yes.
How fast would that be?
I don't know, but then, yeah, it's not short therapy. It's long exposure of your molecule. So,
but then
as
it's
contraceptive, you have to take them continuously, pretty much, or at least let's say once a week or once a month.
No,
but still, it will be long exposure. So at some point, you can actually expect the mutation of the receptor. And there are many cases, as for example, ACE, that's angiotensin converting enzyme. So upon binding of the spike, immediately you have the mutation by the coronavirus. And then another thing is that the spike needs to be processed by the TMPRSS protease. So this is actually a very easy way for the virus to avoid the binding of the spike to the ACE2. A new case could be exactly the same. You develop the molecule, it will be long exposure. So basically the receptor, they will have enough time to undergo the mutation, and then your therapy won't work.
The thing is, with sperm, for example, the Izumo protein is only becoming exposed after the acrosome reaction, which happens in the female tract. So so that's not happening in the male body. So if we are, so the receptor itself is not exposed. So then I don't think it will.
Yeah, but in the long term, basically in several.
But the
After
receptor.
several cycles, actually, of exposure to this drug, actually you can have like the born new mutation, which will actually prevent totally binding of this molecule.
Yeah, but if the binding of the molecule is not happening in the male body, so
It doesn't matter it's male or female.
No, what I'm saying is that the isomoprotein on the sperm is only becoming exposed at the like very close to the event of fertilization, because the protein itself is buried in an internal membrane. So the sperm has two membranes. It has an internal membrane where the isomer resides, and it's sealed from the outside environment. And this protein only becomes exposed to the outside environment in the female body. So what I'm saying is that if it's a male contraceptive, the receptor which I'm blocking is not going to be available to the molecule.
Okay.
There was a question here from a student.
Hi, so really nice talk. I think my question might be an extension to what was just asked. I was wondering, are you possibly threatening people for into a permanent sterilization because of a possible mutation? And I understand that it's still a dream and you still have a lot of work to do, but you said that you did something about coronavirus and normal cells and you stopped the fusion. Yeah, so you already know the possible side effects that you could be looking at when you approach this. Can you somehow quantify whether these side effects would be lower than a hormonal contraceptive? And could you possibly be threatening, let's say, customers to permanent sterilization because of an unseen mutation that would permanently stop the fusion?
Okay, so first of all, in terms of side effects, I would expect small molecules to have far fewer side effects compared to hormones in general. In terms of mutations, I guess that's always a risk with any molecule and any drug that you develop. So yes, we will need to consider that and test for that.
So for example, if it's a viral treatment and there is a mutation, you're in fact curing the problem permanently. So the mutation works in your favor. But in this case, it will become a permanent sterilization of the person.
But I'm not sure I understand how the mutation would occur if the activity of the molecule, if the binding of the protein to its target is very localized in time. So the target is only exposed at a very particular time. Would it still induce mutations? Yeah, I don't know, but we will need to consider that. You're saying it will. Okay.
May I just make a comment, right? That when you take viruses, they do mutate really fast. It's the fastest mutators. And when you take our cells, you know, I guess they look roughly the same as they looked 50 years ago. Okay, despite the fact that I have taken a lot of drugs. And so, you know, I don't have a third hand. So it seems that if the stress is not permanent, you will not have a mutation. You need to expose a living organism to a permanent stress to induce the mutations. That's my perceiving the evolution stuff.
You are testing your ideas on mice. My question is whether this acting of these proteins can be species selective, and I suppose that the target is humans, really. And the second question is that are you not afraid that such a contraceptive is if popularized, could be somehow leaking to the drainage system, generally to the environment, and can stop maybe breeding of all sexually multiplying organisms on Earth?
So the proteins, the Izumo and Juno proteins, which I want to target, they are very different between mouse and human. So I wouldn't worry about making all other animals sterile. That is not a concern because this would be very specific to these proteins. And because it will be specific to these proteins, of course, we need, after the mouse case study, to move to the humans. But we start with mice because, of course, it's easier to do tests on mice. But of course, in the long term, we will need to repeat the process for the human isoforms of the proteins. But we first want to see whether this is at all feasible to interfere with fusion by interfering with the Izumo-Juno binding. So after we hopefully see that that is possible, we move to the human variants. But yeah, no concern of
Do you have any other questions? Yes.
Hi, very interesting talk. So I just wanted to go back to the interaction between Izumo and Juno.
Yeah.
Because I don't know, perhaps you know, and you could tell us, is there anything known about the actual structure of the complex of these two proteins? And if so, do you also have isoforms of these proteins? You know, I'm thinking about personalized medicine, because you mentioned that, you know, you would like your system to work as efficiently as possible with the minimal side effects.
Yes.
And of course, targeting the very specific epitopes of
Yes.
these proteins, of both
Definitely.
proteins,
Definitely.
perhaps, you know, case dependent, could be the way forward. So do you know anything about the structure of these proteins?
Yes,
Can you think
definitely.
of other small molecules rather than just these organic,
Yes,
aromatic
definitely.
compounds that you've shown
Yes.
on one of your slides?
Yes,
peptides
definitely.
and things like that?
Yes, definitely. The
Mhm.
structure has been solved, and very, very recently there was a paper showing also that there are complexes. So on the sperm side, there is a complex between Izumo, SPACA6, and TMEM81. So it's not just the Izumo, even though there is a direct interface between Izumo and Juno. There are other proteins in the complex with Izumo that might stabilize it, might have other effects. So the structure of both proteins has been solved, and also the complex, and yeah, definitely.
So you just need to look for the very unique structure that you could, epitope that you could target,
Yes.
I presume, right?
Yes.
That would be the
Yes,
one,
exactly.
another
Yes,
rationale.
definitely. Thank
Okay.
you. Yeah, definitely.
Okay, this is the last question, guys. I'm a timekeeper. So.
Thank you for fascinating talk. I have a question about, are Zumo and Juno the only proteins that are potentially participating in this fusion, or are there any other candidates that you are considering for this contraceptive approach?
That's a great question. So Izumo and Juno are the only known protein pair which need to interact with each other in order for the fusion to take place. But there are other proteins that are needed. There is a list of proteins, so this slide that I showed, that if you knock out each and every one of them, you will get a sterile mouse. So there are other targets. We just wanted to start from a protein-protein interaction, which is more easy to target. But also, I think that these are not the fusogens based on current knowledge. So it also might be the case that there are other unknown proteins that are yet to be discovered. But specifically, this is the only known protein pair, and that's why I choose to target this interaction. Thank you. That's it?
Okay, thank you. Thanks, Robert, and thanks to everyone for this opportunity. It's an absolute joy to be invited here. And I'm very excited to tell you about, as a synthetic organic chemist, my dream, which I've had for about a decade, focuses on organic molecules that mimic transition metals and how those can be used in chemical synthesis. So I thought it would be appropriate to give you some background about transition metals and why they're even worthy of being mimicked, and what they're going to do in society and how this will hopefully change the world. I'm actually going to give you two reasons. I'm going to give you one right now, and then maybe in about 15 minutes I'm going to give you the other reason. Okay. So I think it's fair to say that society, global society, has a very complicated relationship with transition metals. We are certainly dependent on them for chemical synthesis. This is a survey done a few years ago of the 20 most used reactions in medicinal chemistry. And you can see actually the 5 that I've highlighted in yellow here are dependent on transition metals. So what are these years here with the arrows? These are actually indicating the only 3 reactions that have been discovered in the past half century. So all the new reactions that actually make it into drug discovery are transition metal based. Okay, not only for drugs, but actually we depend on if you have any electronics, or if you use plastics that are based on commodity chemicals, if you use fine chemicals, for example, for your fragrances, or if you eat food, you depend on transition metals all the time. So what is the problem with transition metals? There's a few. Okay, I really like this periodic table. It's colored by scarcity, so economists will like this. Red is the most scarce, obviously, and we have you'll notice a really big concentration of color right in that D block, especially on the right-hand side. That's where the noble metals are that we use a lot for synthesis. So that's bad news. And if we zoom in just on palladium, I'm sorry, this is a very America-centric presentation, but in America, we only produce 6% of our own palladium. All the other palladium comes from imported from other countries. And as stuff starts to get scarce, something that chemists need to realize is it's not only when it runs out that there's a problem. As it starts to get scarce, prices go up and you start to have other kinds of issues. It happens that palladium wasn't sprinkled evenly over the entire Earth, but some countries got more of it. Some of you have seen movies. Others, I'm sure, know, transition metals are unfortunately very toxic. They're not good for you when they go inside your body. They're not good for the environment when they go in the environment. So we spend a tremendous amount of investment in process and engineering controls to make sure that these metals get separated out before use or before disposal of chemicals. And of course, speaking of costs, right, they're very expensive. Right? If you go to a restaurant and you have truffle, try saying to the waiter, okay, I'm going to have palladium instead. Right? Your bill will be an order of magnitude larger. So as I said, a very complicated relationship. So what is it about these metals that makes us actually want to use them from a chemical perspective? Okay, one of the things that metals can do, this is a bit of a simplification, but you can take a strong bond, an XY bond, and a transition metal can go right in the middle, insert right in there. That's called oxidative addition. Okay, and then that's a reversible process. When you want to, the metal can now exit, and you make a bond between those two things. So why is that useful? Well, I can go into a bond, I can swap out this thing, and then I can exit. And that's a process called cross-coupling. And if you look at just some of these drugs that I've highlighted, I could have put 2,000 here, right? These bonds that are highlighted in blue are made using transition metals to do exactly what I just said. Okay, so the idea of replacing transition metals is not new. I didn't come up with that. I should give credit to some really great chemists who have tried to take things like bismuth or phosphorus or carbon, just other elements, and sort of torture them into acting like a transition metal, and that's seen some success. So what we're doing differently is the idea that I'd like to pitch in this dream is to take an entire organic molecule, and here are just some examples, take an entire organic molecule and make the whole thing act as if it's a fat transition metal, do things like oxidative addition, reductive elimination, etc. And I'm going to go into a little more detail about how that's going to happen. But in order to understand the chemical principles behind the design, let's do a little exercise. So imagine all right, close your eyes, imagine you're a molecule and you're about to meet a transition metal. I say, hi, I'm a transition metal. What actually makes me a transition metal? Okay, if you ask most chemists, or organic or inorganic chemists, they'll say, well, transition metals have d orbitals. These are these special things. There's 5 of them. This is one representation. To me, that's only a partially satisfying explanation. It doesn't tell me what about the d orbitals makes it a transition metal. Is it the shape? Does the molecule ask, do you have an orbital of this shape or not? So to really dive a little deeper into this, I'm going to give you a little crash course in group theory. Just kidding, right? I'm an organic chemist. I don't like math, right? So I'm going to go back to this very sort of arcane method of arrow pushing. We've all seen this, right? In organic chemistry, when you have arrow pushing, these arrows represent movement of electrons. And I think, yes, it is a simplification, but it's actually a rather deep thing. So what I've depicted here is oxidative addition, reductive elimination, some fancier processes. This one's called migratory insertion. This one, right, if you read a lot of chemistry, right, this is called CH activation, hot topic. All of these have something in common. So look at the metal that I've highlighted, and I wonder if you can spot what's in common with all of these arrow pushing mechanisms. Okay, so what I think is in common is all of these have an arrow that's going to the metal and an arrow that's coming out of the metal at the same time. And I think that's what makes a transition metal. Okay, so I'd like to propose this. A transition metal has something called bifelicity. It can act as an electron donor and an electron acceptor at the exact same time. What does that mean in terms of orbitals? Okay, if you want to be a good donor, you need an occupied orbital that's pretty high in energy. You need electrons that are ready to go, they're very high in energy, and you can donate to various kinds of acceptors. Okay, the exact reverse for what makes a good acceptor, you need a low-lying unoccupied orbital so that other electrons will want to go in there. And if you want a high energy filled orbital and a low energy empty orbital, what that means by arithmetic is that the distance between your HOMO, the energetic distance between your highest occupied molecular orbital and your lowest unoccupied molecular orbital, that has to be a very small gap. So that's one consequence. The other thing is these frontier orbitals can't be all the way up here, okay? You can't be too metallic or you can't be too non-metallic. You kind of have to be moderate in terms of redox potential or electronegativity, whatever you want to call it. Oh, okay, one more thing, by the way. It doesn't help if the LUMO is here in Warsaw, okay, and the HOMO is in the Czech Republic, right? They actually have to be overlapping. They have to be in the same place in order for you to react with the molecule. So, yeah, you found my LUMO? Yeah. Okay. So let me put that into more fancy chemistry jargon. My dream, part one, is to find an organic molecule. First, it has to have a similar redox potential to transition metals. So it has to do redox, just like if it were palladium or iron or whatever. Okay, and it has to have a co-localized highest occupied and lowest unoccupied orbital with a very, very small energy gap. If we can fulfill all these requirements, okay, my dream is true. So for those of you in the audience paying attention, right, if I say I want an organic molecule that can do redox, well, we already have those. If you, in photosynthesis, right, in the electron transport chain, or if you've read the redox flow battery literature, there are organic molecules that can actually store electrons reversibly. And these quinones are one class of them, and the way they work is, imagine you have, this is called orthobenzoquinone, and when we add 2 electrons and 2 protons, you go into this reduced form, and that's reversible. You can take those electrons in and out. And this dream really originated when I looked at this, right? You squint a little bit, and you see it looks exactly like if you take a transition metal and do oxidative addition to dihydrogen. Okay, and so the question is, can we use that analogy in real life? Because this analogy, as it's drawn right now, is just poetry. It's something that I wish were true, but if you add hydrogen to orthobenzoquinone, this doesn't happen. The thermodynamics are right, the starting and end points are okay, but the barrier to get from A to B is just way too high. Okay, so how do we satisfy the kinetic criterion? Okay, I'm going to draw one roadmap, would be to replace this oxygens with sulfurs. Okay, we all know that sulfur is more nucleophilic than oxygen, it's just bigger. And also, a carbon-sulfur bond, okay, a carbon-sulfur bond is less polarized than a carbon-oxygen bond. They have about the same electronegativity. So now you have some acceptor character lying on that sulfur, whereas for a carbonyl, for a CO, it used to be all on that carbon. So all of that stuff put together just means that this resonance structure, this kind of ambipolar resonance structure, is a bigger contributor when you have sulfurs there. Okay, so my hypothesis is that if we look at these dithioquinones, we will get enhanced bifelicity and therefore maybe metal-like reactivity. Okay, very little is known about these compounds, and it's not trivial necessarily to make this happen. For example, we know that these compounds can exist in equilibrium with 4-membered ring forms, but we do hypothesize that if you have electron donating groups, you can favor the open form over the closed form. There's also other bad things that can happen. They can oligomerize, right? Sulfur likes to bond to itself, or you can, actually have destruction of that CS bond. But there are always, design strategies that can help you prevent these things from happening. And assuming we can do all that, if you just take a model molecule and do a very crude calculation, you do see that you have HOMO and LUMO character lying on sort of those same atoms, as particularly the sulfurs. Okay, so let me just show you one reaction that we plan to do. If you take, I'm just drawing designs, out of my mind as an example. Take this and imagine you add an aryl chloride, okay? Imagine this sulfur now comes and takes this arene group, and at the same time, the other sulfur comes and picks up the chloride at the same time in a sort of concerted mechanism. You can split that carbon-chlorine bond across this, and this looks exactly like, oxidative addition of a transition metal. Now, we're going to take that chlorine and swap it out for something useful like an amine, and then we just want the opposite of step 1 to happen, and then now we've made sort of that CN bond. So this is just sort of one example of something that I dream could be reality if we made an organic pseudo-metal. And there's all kinds of other design principles you could start dreaming of. Maybe we want to put some substituents here to push those groups together. Like I said, maybe we want electron donating groups to disfavor this form, et cetera, et cetera. But these are all really details. What we want is an organic molecule that as a whole acts like a metal. Okay, so if I were to draw a rough roadmap in the lab, we would start by synthesizing some dithialquinones, because really not very many examples are known. And then we want to find ones that can do this process, okay, break an XY bond. And we want to make some of these and find ones that can make an XY bond. And hopefully, what I'm hoping is you have the same scaffold that can do both, and suddenly we have catalysis. Once you have catalysis, then we're in business. We know how to optimize, and we know how to make different reactions, extend that chemistry, et cetera, et cetera. And instead of talking about optimization and extending that chemistry, I promised you a second reason for doing this, right? So far, the reason for doing this has been the societal need for things that do what transition metals do, but aren't transition metals. There's also, for me, a more exciting reason, which is the intellectual opportunity. And to do that, I have my friend here. So raise your hand if you know Mr. Potato Head. Okay, so this is an American icon. Okay, every American child has one of these in their bedroom. The concept is Mr. Potato Head is a plastic potato. Okay, there's more. Right, the plastic potato comes with different eyes, different ears, different nose, right? And you just put every possible combination is equally ridiculous. So you just choose some and you put them together. So let's imagine Mr. Potato Head asked ChatGPT, okay, just give me 10 metals, and then also give me 10 organic reactions. Okay, I think you know where this is going, right? Then we just take each of these and pair them together. Okay, and I'm going to go to my friend Google Scholar. I have lots of friends. I'm going to go to my friend Google Scholar and ask, how many times has that exact phrase appeared in the literature? So what do you predict the results will be? Okay, so I actually got lucky. I found one reaction that hasn't been studied. So if you're a PhD student and you're looking for a thesis topic, right? But in general, what my point is, this is a 0th order approximation. It is a little bit crude. Chemists are so capable, and there's been so much labor put into organic methodology that pretty much for every metal, every class of reaction has been looked at. And our creativity and imagination at this point is actually somewhat limited just by the fact that the periodic table has a limited number of elements. And so what I'm thinking is, if we go into this business of mimicking elements, right, why stop at mimicry? We can actually imagine we're basically synthesizing new transition metals that don't just copy palladium or copy rhodium, but they might actually have new properties. Okay, so for example, one design might one day end up as one of those entries, and we could have many more. So in other words, my dream within a dream, okay, is to have not just mimicry of the important metals that we need to replace, but actually to look at new opportunities and new reactions completely inaccessible to the current conventional periodic table metals. Okay, so in the next few minutes, I'm going to give you maybe 3 examples of some new opportunities that I think are exciting. They're just examples, and then I'll wrap up with sort of an interesting food for thought, okay? So one thing we could do is we could take a transition metal and look at what are its limitations. What do transition metals not do very well? And one of those things is they can do so many things, sometimes it's hard to select which thing it does. If we look at this structure, this is just an intermediate in a CO coupling where we want to make a bond between this aromatic group and that oxygen. So we want to go down this bottom pathway and make that red bond, okay? Now it happens that when metals have a hydrogen exactly 1, 2, 3 bonds away, that's called the beta position. And when you have something in the beta position, it can do beta hydride elimination. So that metal just picks off that hydrogen, you end up with hydrogen there instead, and then when it eliminates, you get an aryl H bond. Disgusting, no one wants that, right? So my argument is, if we're gonna design something that acts like palladium, we don't have to turn on every possible reaction that palladium does. We can turn on the ones we need. We can add to an electrophile. We can form the CO bond that we need, but we can leave it, intentionally with no vacant orbital here to actually, pull off that hydrogen. This is just an example of, you select off the menu which processes you want to design in, and you don't necessarily have to be a victim to the other ones. Okay, another thing we could do is we could do the same reactions as palladium or another metal. We could do the exact same reactions, but with different selectivity. And selectivity is really crucial in organic chemistry. So if I bring you back to slide 12, you might have noticed reductive addition, oxidative elimination, right? It's like, Richard, did you have too many drinks last night? That's not what they're called. They're called oxidative addition and reductive elimination. But there's a little quirk to the systems that I'm showing here, which we can discuss in the question time if you want. If you look at this transformation, even though it looks like an oxidative addition, on the red part, it's actually net reductive. So what happens here is when you have an organic system like this, for certain chemical reasons, it has the opposite polarity of how transition metals typically react. And that means, suppose you have a molecule that has an electron-rich carbon-chlorine bond and an electron-deficient carbon-chlorine bond. Well, if you were to just throw in a normal transition metal like palladium 0, it would definitely add to the less electron rich position because it's doing an oxidation. But if you were to use, for example, the kind of organocatalyst that we're proposing, maybe you could actually get selectivity for the other position by virtue of it being a reductive process. Actually, a corollary of this is not just selectivity, but if you take bad substrates for traditional cross-coupling, they could actually become the very best substrates when you have a new type of metal that is designed sort of in the way I've proposed. Okay, so let me just give you one final example. Okay, I think it's pretty uncontroversial to say that if you have a transition metal, it's more or less a sphere, right? Just like the Earth, although in the US it's kind of controversial to say that now. When you have a spherical thing and you have two really large groups you're trying to couple, sometimes you just can't fit two very large things on a sphere. It's just a single point, okay? And that actually turns out to be a real problem. For example, people, to my knowledge, have never done a coupling with this group SF5. It's just too big. You fit it on a metal, stuff starts falling off or it starts decomposing, etc. That's just because the metal is a single ball. Now, if you start designing things that are designed to mimic metals, you don't have to make it a ball anymore. It could actually have independent binding sites. You could actually control, this binding site's more electron rich, that one's more accessible, sterically accessible, this one's less sterically accessible, etc. So now you sort of just craft these groups in a way that you can have small groups up here fit an SF5 group or a CF3 group, etc. And there's all kinds of cross couplings that we can maybe realize that you can't do currently with your sort of spheroid conventional metals. Okay, so these are sort of three examples of new processes, and I could give you many, many more, but what I wanted to do is leave you with this very interesting, I think, very interesting thought, which is we've talked a lot about oxidative addition and reductive elimination, but I want to propose this statement. Actually, every transition metal catalyzed process, every transition metal involving process you can think of, I can draw an organic analog using an organic molecule to replace that transition metal. Okay, just as one example, if you had a metal dihydride, you could imagine, theoretically, if you react that with carbon dioxide, this is a balanced equation, you could eject water and make a metal carbonyl. And you may know that metal carbonyls, if you shine light on them, a lot of them will just eject that carbon monoxide. That carbon monoxide comes off as free CO, and you just get metal with its other supporting ligands. Now, if you just add dihydrogen or reduce it in some other way, you could get back to a hydride. Okay, so what we could do is we could take an aminothiol, for example. This is just an example structure. You see these blue hydrogens sort of correspond to that hydride here, and we just condense on carbon dioxide. This is actually just a condensation reaction like you'd see in first semester organic chemistry. And now, when you shine light on something like this, it can actually undergo a pericyclic reaction where it just ejects that carbon monoxide, and you get this thing resembling a quinone like I showed you in the beginning. And if this could activate H2 or otherwise be reduced back to the original cycle, then if you could have this all happening in one pot, I'm not promising that this can happen, but this is just an example of how you can design reactions using this general concept and maybe come up with, dream part 3, part 4, part 5, et cetera, et cetera. But for the sake of time, I will just leave us on that note. I want to thank Robert, Pavel, Adam, everyone again for inviting me, and I'd be happy to have some discussion about this proposal if you have any questions.
Thank you, Richard. How stable would be your organic catalyst?
It's really unknown. It's actually surprising. I talked about metal reactions that are unknown. Organic functional groups that are unknown is also a very rare thing, but this thing, to my knowledge, hasn't been isolated or characterized. What we can do is anticipate some of the ways it can decompose. One of the main ways that thiones decompose is just by moisture. It hydrolyzes the CS bond. We can, to some extent, block the carbon from doing that. But the fact is, maybe this is cheating, but I'd like to say even if it is sensitive to water, transition metals can be sensitive to water as well. So we can't solve every stability issue, but I do think that these compounds deserve a further look. Historically, when we've looked at compounds that have never been made before, we've always found interesting stuff.
Okay, so let's assume you find a catalyst which is free of the transition metals. But does your approach also includes design of the reaction pathway to synthesize this catalyst without any transition metals?
Yeah, I do apologize that in this talk I haven't put in a synthetic route. We have, as you may imagine, tried actually to make some of these compounds and had some success. Can't go into specifics right now, but I think sulfur compounds are very underexplored, and people assume because of that they're extremely unstable and hard to make. they're not that difficult to make. It's stinky. I mean, it's not fun to work with sulfur compounds, but it is fun in the sense that you can make a lot of them, and we've actually discovered some related functional groups that hopefully you'll see in the literature sometime soon.
Do you have to use the catalysts with transition metals to synthesize them or no?
Okay, that's actually a really good question. On discovery scale, sometimes that is the easiest way to synthesize a library of compounds. To prepare them, we all have, at least in mind, transition metal-free ways to make them. But even if you had to use a transition metal to make a catalyst, if that catalyst can now be turned over 10,000 times, you're still cutting down a lot on the transition metal. Plus, if that catalyst is used in the last step of a drug synthesis, you'd rather, the transition metal be one step further removed than the transition metal actually being involved in the last step of your drug synthesis. There is a subtle but very important difference. Thank you.
Questions?
Thank you very much. Very nice talk. With the parameters that you discussed, you were controlling the splitting of the bond of the substrate. But at for this compound to act as a catalyst, at certain point you have to release it from the catalyst. Have you considered the parameters that are responsible? Because this bond can be later quite strong with the sulfur.
Yeah, I understand. Maybe I should. Sorry for doing this. Close your eyes for 2 seconds. Yeah, no, I understand exactly what you're saying. The tighter you make something bind in this step, the harder, obviously, the reverse step will.
Like the structure on the right. This can be quite stable structure, for example. Then your catalyst is inhibited.
Right, but the great thing about these is like tuning a ligand on a metal, you can actually tune the binding and the relative stability of this structure versus this structure. Obviously this structure has some things going for it, like it's aromatic, but also, sulfur, chlorine, and sulfur carbon bonds are not super strong. So all I can say is I can't predict which structure will win, but I can promise you there's a pretty high degree of tunability here. I can make structures that will never do this, and I can make structures that will only do this, and the question is finding that balance.
I'm just wondering if the same parameters that you considered for splitting are also important for the...
Oh, I understand. Yeah, so the thermodynamics of the process are not as controlled by those orbitals, because those orbitals change when you go from one state to another, and it's the change in those orbitals that controls the thermodynamics. What that homo-lumo splitting controls is the kinetics, how reactive, how low is that barrier. And to some extent, at least in a design principle, we can decouple the thermodynamics from the kinetics, I hope. But yeah, it's obviously a great question, and something that computation, I hope, can help with. I'm not an expert, but I have friends who are, and I think they would be happy to collaborate.
Okay, thank you.
Okay, great talk. Do you think that dimerization could be a problem for such catalyst?
Yeah, I do think that, when we tried to make some of the very simple ones, that is an issue. Dimerization and further oligomerization. What's not clear from this 8-membered ring diagram is that ring's actually not flat. So bond angles at sulfur are closer to 90 degrees. So this is actually kind of like and because of that actually makes it a little bit easier to inhibit the dimerization by just putting really fat groups next to the sulfurs. Because it's not flat, you can actually kind of thermodynamically disfavor that. Does that mean I can show you a structure of a monomer? Not right now, but I'm very optimistic. I'm dreaming. So.
Could selenium replace sulfur to make it less stinky and maybe a better catalyst?
Better catalyst, yes. Less stinky, oh boy, I don't know. It's a matter of opinion, right? I actually prefer the smell of selenium, but I think my lab members do not. It's actually pretty bad. And selenium has its own toxicity concerns at a certain level, and sulfur tends to be relatively benign. So we are definitely looking at selenium. You can imagine putting other elements in there, phosphorus, nitrogen, anything. But it happens that the space here is so big that we kind of just had to pick one to explore, and sulfur was my choice. No real reason other than I love sulfur.
Thanks.
Okay.
Fantastic talk. I have a question about other scaffolds. Are you considering any other sulfur-free scaffolds that could do the same job?
Mm-hmm.
I know that probably you don't want to go into specifics, but
right.
How many ideas do you have? Can you tell us a little bit about it?
I haven't actually gone and counted the ideas. I will say I put a structure on here that is gives us something concrete to look at. They're not, of course, the exact structures I'm working on, but I think for the purposes of discussion, good enough. I think one of the beautiful things about this kind of chemistry is you start discovering stuff by accident. And to give you some idea of other scaffolds, quinones are not the only, we take a lot of inspiration from biology. Quinones are not the only redox couple in biology, and so there's others. There's a question of, do you want to look at stuff that contains metals, but the reactivity isn't happening at the metal? The metal's just sort of helping you. From an intellectual perspective, of course, those are very interesting. From a perspective of replacing metals, obviously it's different. But I think our lab is fairly fundamentally driven, so we are looking at other systems as well that involve metals, but just as a helping hand and not actually doing the hard work. So that gives you some idea of other scaffolds.
Other questions, comments?
Your design is two-dimensional, and transition metals complexes, they tend to be three-dimensional.
Mm-hmm.
Can you add a third dimension, like anchoring sulfur to C60 or something like that?
Yeah. The challenge is, the answer to your question is yes. The challenge in terms of design is you have to keep it electronically conjugated while also having the 3 dimensions, and then you run into this problem that carbon only makes 4 bonds, and then it becomes hard to design things that are sp2 hybridized but have 3-dimensional. It's not impossible, and there's definitely ways to do it. What I will say is a lot of metals in their catalytically active redox states actually also do adopt planar geometries. So what you do is you put ligands on there, or even worse, they can be linear geometry. So you have to put really very specialty designer ligands in there to get sort of a 3-dimensional structure around your active site. This just takes the metal out of the equation, and you still have the same design considerations, but your organic thing is interacting directly.
Just a pretty blunt question. It's a very beautiful dream, but from the talk I didn't get if you're still dreaming, or do you have any proof of concepts, some reactions that you performed that actually show some promise?
Let's just say I'm still dreaming. I mean, of course, the hands are dreaming as well, and there are things that make me more optimistic about this than when I first thought of it.
Okay.
But this is not it's not a done project pretending to be a not done project. So if you're asking
that's
about
completely fine now, because this is like a really beautiful dream. I like it.
Okay.
I was
I
just
appreciate
wondering if all the reactions that you have shown are just a paper.
No. I mean, it's a dream I and my students and others believe in, so we are definitely trying it out. Yeah, among other things. Yeah, thank you.
Thank you.
When you do organic chemistry, you synthesize small molecules.
Mm-hmm.
There are a number of rules, roughly around 100,000.
Mm-hmm.
It's a rough estimate. Which of the reactions are possible or not possible.
Mm.
But you can make it algorithmic, right? Once you make it algorithmic, you can teach AI how to do it.
Mm-hmm.
You may be out of business before you realize your dream.
Mm-hmm.
Convince me that you will be not.
Okay. Well, first, maybe I don't want to convince you, because I would love to be out of. I mean, I don't care if this dream is realized by me or someone else. That's why I'm here telling everyone. But I think right now AI for discovery is an extremely promising field in organic chemistry. But because of the limited amount of training data that is reliable that you can give it, sometimes when an AI claims to make a discovery, the problem is there's no way to run a control experiment where a lab would have worked on the same problem without AI, and you see whether they would have discovered the same result. And so, am I going to engage with the AI community to try to do something like this? Of course I am. Is it going to replace what my lab is doing in the meantime? I don't think so. And I would love for either of us, in a very friendly competition/festival way, right? If anyone could discover the answer to this problem, I think it would be really big for all of chemistry, and it would inspire other groups to work in this area as well. And I think that's a great thing. So if someone else wants to beat me to the punch here, I would welcome them to do it.
Okay, so maybe I will reverse my comment, okay?
Okay.
You are too good to be out of business.
Okay.
But how are you going to collaborate with AI? Because you
Mm-hmm.
will have to collaborate if you don't want to be out of business.
Right.
Okay, so a trained chemist on the top collaborates with AI.
Mm-hmm.
How would you do it?
Yeah, I think probably the same way I would collaborate with anyone else, I would first ask them, what do you need from a chemist that you can't provide by yourself? And I think, hopefully the answer isn't nothing, right? Hopefully the answer is, here's a list of stuff. And, human expertise can really still right now play a pivotal role. I mean, there's things, let me put it this way. There's things people would like to discover in the short term, much more important than this. People want to discover drugs for cancer, et cetera, et cetera. They're all working with AI. But they also need their human expertise. And so I would model my discovery efforts after that. I would figure out the things that I can do best that they can't, and then give them here's a guide. Maybe don't look at these compounds. There's some rules. We figured out we can't make these compounds, et cetera. So just keep updating your models, and then likewise I encourage them to come to me at a very early stage. Oh, my computer is suggesting this thing with 18 seleniums. Can you make it? I would love to give them a little bit of help there.
Any other questions or comments, or we have a coffee break? Yes, you vote for the coffee break now. So let's thank Richard once again.
Okay, all right. So good morning, everyone. My name is Zach, and I'm super excited to be here today to share my passion and a vision for the chemistry that inspires changes.
Recording in progress.
So, but before diving into the science, let me first start with my personal story. So, okay. All right. So in my free time, so I'm a photographer and a hiker. So I remember, visiting Europe for my first time in my whole life, which was back on 2019. So the first country I explored was Iceland, a place, full of stunning glaciers. So I will never forget the moment I was, standing on this, vast glacier, and I'm feeling so tiny compared to the nature's ground scale. So actually, in the middle of my trip, I asked the guy that helped me to tour this glacier hiking. I asked this question: is this glacier ever growing? It is so large. So how large can it actually get? And to my surprise, he kept silent for about like 5 seconds. And then, so he showed me this photo and told me about 30 years ago, it looked much more different. So the glacier, as you can see from this slide, had diminished significantly since the time he started his job, and it just kept diminished over the next 30 years. So I was actually shocked, and then I asked myself, what will it look like 30 years from now when I'm on his age? Will the next generation of people see what I saw on that day? And sadly, this wasn't an isolated experience. So, this is not just glaciers, but also many other places are changing very quickly. As a landscape photographer, I have heard this kind of story over and over again. So no matter which place I go. So take, Colorado River as an example. It is actually getting worse and worse every year, as you can see from this picture. It is shrinking, and the amount of water is actually running out. So here is a photo of the Reflection Canyon in the southwestern of the United States. So for some of you who has been able to visit, like in terms of like state like Utah, the southwest of the United States, so you may be familiar with this kind of beautiful red sandstone landscape. So this place was actually named for the way that water once just beautifully mirror the sandstone here. And it used to be a place just full of water, so you can clearly see the reflection, in the daytime. So this photo was actually captured back on 2014, and I got this photo from the National Park Service website. So last year, I hiked to the same spot, and this is what I saw. So almost no water left. So I took this, with my iPhone video. So it is actually really heartbreaking to think that in the next 5 to 10 years, this place might just lose its name of the Reflection Canyon entirely because there's just no water to reflect. So what is even worse is that the story of the Reflection Canyon is just one representation of the broader water crisis impacting the entire Southwest United States, where many areas are just crying and suffering with the seasonal water shortages. Okay, so now we are done with the sad news. And then let's talk about something, more about promising in the future. So as a chemist, I always think at the molecular level. So what is causing this, and what can we do? How do we ensure that a future generation can enjoy the sustainable lives on this planet? So even small climate changes, as you can see from the previous slides, can actually greatly impact vast landscapes as well as the critical resource that we human beings rely on. Fortunately, so we chemists deal with small molecules every day, which, despite their size, can actually have a massive impact on the climate. So our challenge here is to actually find a way to control, these gas molecule I show you in this slide, which means either by capturing the harmful ones or generating the clean alternatives. So these require us to think about, trying to build about like atomic level factories that is capable of handling this large volume of gas molecules. So, one promising materials that was proposed, a while ago for this challenge was metal organic frameworks, which is abbreviated as MOFs. So, this material is actually known for their extensive internal structure. So imagine that, you can think of this material as the intricate Lagrange models, where, you will have the inorganic nodes, which is made of the metal ions or the clusters, all these, organic linkers, which bridges these, clusters together. So if you look at this yellow region, which is the cavity space for this material, you will realize that compared with the volume taken by the framework themselves, so the central pore here is so spacious that, it can hold a huge amount of gas molecules. So to give you an idea of, the scale, so 1 gram of the typical MOF can have a surface area which is equal to the entire football field. So think of them as the, the crystalline sponges, which in the end is on the microscopic level, and it makes it very ideal for handling the gas molecules. So in another example I show you in this slide, so we have another MOF with a smaller surface area, but still very large, which is equivalent to the 3 basketball field. So as you can see from this video on the left side, so which I'm starting to play in, so essentially, for this specific MOF, so it is bridged by this, dicarboxyl linker with a nitrogen atom. So these nitrogen atoms serve as the specific adsorption site to attract the first water molecules. And then the subsequent hydrogen bonding further takes more water molecules into its pore. And then finally, this MOF is actually saturated with tons of water. So it is the ability to engineer the structure at atomic level, by introducing different atoms and, to change their arrangement that make it possible to tune the MOF's property for different gas capture and storage applications. Then, so you will be sure asking, hey, Zach, so in the past 2 slides, you have been showing us very good materials. Why don't you just go ahead and use them for the gas capture and clean water applications? They look so promising, right? So here is what the current limitation we are facing. So for the gas capture and the storage application, they depend heavily on the design of the pore environment. While some building blocks, work really well for some specific application, like water harvesting in the previous slides, others, may need to be redesigned for the other application like CO2 separation, methane storage, and so on. So at the end of the day, each single time you need to come up with a different building block, and which is combined with a different metal and a different organic linker to create a specific pore environment. And it relies heavily on this kind of chemical intuition for the design of the building block. And there is almost no guarantee that the building block you choose can actually end up with the best for the application you aim for, which end up with being a lot of trial and error approach in terms of the designing of the framework structure. And then the question will be, instead of just using one component, can we actually try to think about create a multi-component crystalline frameworks that with the customizable properties by just varying their composition? For instance, we can try to change the chemical interactions or just adjust the pore size by selecting different organic linkers. At the end of the day, just like like a Lego factory I show you in these slides. So you can try to just have one single platform with multiple units to function in a different way for different tasks. So more importantly, you can make this kind of cycling between the absorption and the desorption of the gas molecules very energy efficient, because for the crystalline materials, as you can see on these panels, so they are typically have this kind of step in their isogens, which is really sharp. While in other amorphous materials, you are most likely to see the more like shallow and gradual increase of the uptake over the relative pressure. So what does it mean? So it means that the advantage here is that when you use the crystalline materials, so you are actually can have the less changes in when you are doing the pressure or the temperature swing. For instance, let's say if you are trying to change from this high uptake mode to this low uptake mode, so actually you just need to vary a very small amount of the pressure in between to make the mold to evacuate all these kind of gas molecules. And then in this case, so when you have the multiple building blocks, so the behavior of this gas molecule inside the crystalline materials will be more precisely controlled. So in some cases, you can even try to think of reverse the selectivity by using the different compositions. Now, instead of trying to redesign the building block and do all this kind of trial and error again, so you can try to achieve this with a single platform with the multiple building units. And then, besides of having the advantage in terms of the separation, so you can also try to have different metal ions, which can make the platform really good for the cooperative catalysis. So the resulting structure will have kind of complicated pore shape with different kind of reactivity because of the heterogeneity in terms of the metal ions. And just like the enzyme, so you can allow them to selectively binding of the specific molecules. Taking all these kind of advantage together, so we can envision that this will result in a more versatile catalyst platform compared with their just single component counterparts. And now, so what will this dream material actually look like? So just think of how our Mother Nature, creates the sequence and the folding of the protein, where a certain number of the building blocks is vary in different way to create so many different possible functions. Similarly, we can try to build up such artificial crystalline frameworks with the programmable structures by incorporating different organic and metal building blocks. In this case, so as I mentioned before, so the ultimate goal here is that we want to engineer those cavities inside the materials by introducing the heterogeneities, but make sure they are still in a periodic manner. So in one unit cell, so they may be different choice, but when they form this extended structure, so the properties can be precisely controlled by us. And these complex pore environments will allow broad applications. So consider the chemical separation, which actually accounts for about 15% of the global energy consumption. So by using these mixed linker and mixed metal microproduct structures, so we can try to optimize the processes like separating hydrocarbons in the petroleum industrial. Additionally, we can target the greenhouse gas and the pollutants removal by enhancing the binding affinity, and also try to create the high capacity materials for the clean water and clean energy applications. Now, I have been showing you, some great promising ahead of us, but of course, there are always challenges. So in fact, how to synthesize such complex materials has always been a problem. So you may recall from your high school or college teacher that during the last century, it took many years for the scientists at that time to develop the pipette synthesis. So peptide synthesis. So and similar challenges is also facing for the MTV MOFs. So let's say you want to synthesize the MTV MOF with just 3 metals and the 3 linkers. So in an ideal world, so you mix them together and expect, to form this, a perfect product, which all of them, just come together in one framework. However, chance exists that some of the linker just, all the metals just didn't get incorporated, and then you end up with this two-component mixed linker, mixed metal system. And it could also happen that, one of the linker get incorporated, but the rest two are just missing and just hanging around in the solution. And in some case, you may just end up with only one single component morph, while, the rest of the two metals and the two linker just don't want to assemble into the structure. And what is even worse is that you actually have hundreds of possibilities, and how to precisely synthesize the one you are actually looking for really becomes a problem. So even with just a few components, so in this case, which is the number is 3, so the number of combinations at the end of the day is so vast. So simply just mixing components in one round bottom flask or the vials is unlikely to result in the self-assembly in this case. Instead, so we need to really think about the precise crystallization conditions for the exact one formulation, we are looking for, and try to figure out, how we can achieve that specific composition. So then you will really need to think about, what conditions you will use in the wet lab synthesis for this crystallization. So there is an old Chinese saying, which is about knowing is easy and doing is hard. So both, computationalists and experimentalists can, design the numerous materials. But the hard part is always to go to the wet lab and actually make them. So the consideration in this case, include but not limited to concentrations, temperature, reaction time, and the solvent. So it is, as I say, it's straightforward to design the materials and run the simulation. However, so the currently the gap is that how we can actually, synthesize that and figure out the exact condition for these materials with the promising properties. So I am trained as a synthetic chemist, and my experience told me that this is not an easy task. So you will need to pick up the exact right conditions so that, they can crystallize into the thermodynamic stable form. And one long-standing challenge, as I say, in this field is that there are just so many choice of the synthesis parameters. In this case, you need to think about. And in this case, for a 4-dimensional space with 4 different synthesis parameters, let's say each one, if you have 5 choice. So if I do the math correctly, you will need to screen for about 600 reactions. And then you may say, well, let's just try to do the high throughput screening, or then spending 2 months trying to run all these reactions. It should be, like not that kind of laborious task. But what if, you want to think about the fact that we are actually talking about a multi-component morph where you have 3 metals and 3 linkers. Now you have much, much more choice you need to worry about. And even with the design of the experiment, still it's a quite labor-intensive task. And you may kind of use your chemical intuition to come up with, an educated guess for the initial condition to start with. But, at the end of the day, it's very similar to have a lottery in the United States. So the chance is really low for you to get lucky to be there. So the key here is that for such vast space, which is actually beyond the capacity of the traditional trial and error approach. So what should we really do? So since we are, dealing with the programmable materials, we are trying to see if we can program their, sequence and also composition for the specific application. So why don't we just try to use AI to actually program and guide this development process? So first of all, we can think of develop an AI model to actually read and mine the literature data, especially those ones with the crystal structure and the synthesis conditions. And second, we can try to think about to use the obtained data to actually train a foundation model that is capable of predicting the framework structure, which in this case, I will just call alpha morph. So just like, you can try to input the protein sequencing to get a prediction in terms of the 3D structure. So now you can use, metal and the linker, and as well as, the synthesis conditions to predict, how they will eventually connect in the extended crystalline structure. So the model will actually also tell you, what will be the favorable topology at the end of the day, which is essentially the folding analogy of the moss. So, at the end, the most important advantage that chemists can use this model to actually screen is that they can use that to screen for the promising candidates without the need to run a single crystal X-ray diffraction analysis, since you can just predict their crystal structure. And then you can just input those, building blocks and synthesis conditions to just output the crystal structure to examine them easily. And finally, once we figure out what kind of structure or the materials we want to make, so we can also have the autonomous synthesis with a feedback loop to actually enhance the materials crystallinity and other properties, which can be done through many iterations of improvements with the machine learning model, which can be used to guide the synthesis and dynamically adjust the conditions to effectively help chemists to navigate this vast synthesis space, as I showed you before. In this case, so we can collect data on both success and failures, and then try to make improvement not only by knowing the good data points, but also learn from those failures. So here, I'm showing you like a baby version of the demonstration on what features this kind of AI model will look like in the future. So a chemist can be able to just simply input the in this case for the morph prediction, the metal and the linkers, which can be done in terms of the multiple metal or multiple linkers. And then the chemist can try to include the synthesis conditions for screening purpose, such as temperature, concentration, and the reaction time. So since this foundation AI model is trained, will be trained on the both crystal structure and their synthesis conditions from the experimental data. So for these given initial conditions, so it will make prediction on the likely resulting crystal structures and also give you some properties associated with these predict crystal structures. So in this case, you will be able to see, if, you have similar structures that has already been reported, or, the AI model will believe that you will actually get a novel morph structure. Either it's novel because it has a new topology, or it's based on an existing topology, but have a different, compositions. So you can try to, download like a 3D crystal structure of the atomic, coordinations to examine it further or compare it with the existing structure, for their properties, which is stored in the database. So since now you have a safe structure you can look at, so the model will actually also help you to predict the properties such as the gas sorption or the stability by connecting, the existing 3D structures with the computational packages or simulation softwares. And of course, this interface is for the simple tasks, like you just have one or just multiple candidates to search and easily predict. And in practice, so the power of such model really lays on the fact that you can easily scale up this entire screening process by just setting like a threshold or like a specific target, and then you can just easily screen for thousands or even millions of combinations, and then at the end of the day just systematically analyze all the predict structures. So which in essence is the digital synthesis of the framework materials. And you will be much, much more efficient compared with, trying to designing the structure for the building block using the empirical knowledge and then try to just enumerate all the possible combinations. So I also want to note that, this interface is just a future outlook, and we are not there yet. And, getting enough data to make this kind of model to be accurate always requires the effort from the entire community. So there has been a very decent amount of experimental morph data in both the literature and the CCDC database. Yet a majority of them, are not readily available for the model training and can require a lot of annotation and the modification in terms of, cleaning the data. So, but I'm very, overall, I'm very optimistic about this bright future as more and more people are starting to reporting their, data points, regardless of success and failures in a more comprehensive and detailed way. So that, there will be more and more useful data for the training purpose. And then, so once you find some, promising candidate from this screening and prediction process, so let's say, you want to move forward with the synthesis. And then, so there will also be another AI model for you to help to navigate this complex space. So a typical challenge that morph chemists, facing is usually, it takes a long time to find the optimal crystallization conditions or, growing their single crystals in order to maximize the porosity and the performance, since there is always like a correlation between like how, crystalline your materials are and how, how porous it end up with. So if the compound is not, highly crystalline, sometimes it doesn't reach its maximum theoretical porosity. And in this case, its properties is diminished compared with what is predict. So in this case, in order to navigate such different synthesis parameters and screen the best ones, so we can incorporate an active learning approach to statistically drive this optimization process in a more productive way. So in this case, even though you are not an expert in the morph synthesis, so as long as you know what kind of objective, for instance, like the crystallinity, the yield, the surface area you are aiming for, so you can easily just try to get suggestions from the machine learning model, and then a lot of time it will be much more productive than the approach of just varying one factor at a time, as I showed in the previous slide, or for this kind of crystallization optimization. So essentially, just to be more specific, so the way this, synthesis optimization model work is that, so after testing a certain number of synthesis conditions, let's say, if you are running 5 initial reactions, so you will need to measure their properties such as surface area, and then so try to feed back the desired property data back into the model, which, the model will be able to make suggestions for the next round of the experiment. So this iterative process allows AI to learn from both success and failures in a real lab environment, and then try to make its predictions increasingly reliable as you do more and more experiments. So what will even be more exciting is that all these, 3 AI models can actually be integrated together, and we can actually automate this entire process one day in the future, from reading the literature to predicting and synthesizing the materials. So here, my biggest dream is that, one day we can just walk into a lab and then tell AI assistant something like, I will need a material for gas separation between, gas molecule A and B, and then you specify some operational conditions such as 400 K, 2-bar relative humidity of 3%, so far and so forth. And then, so it will give you, many different proposals, and you can just work with AI assistant to examine this proposal and, ideas, and then just pick one. Then this self-driving lab will just handle the rest of the test. So after a few days, you just walk back, and then so you can just, in the days between, you can relocate your creativity to some other stuff, while, saving time on the most, tedious and routine laboratory part. So when you come back to the lab, so you will have a new customized materials, which has been optimized for your needs. So this is actually where the tunability of this multi-component framework material truly shines. So you have just one single platform. And then the question is just how to choose and assemble different building blocks, rather than, trying to redesign these different building blocks from the very beginning. And then, so I'm confident that, by dramatically shortening the time, especially this cycle from the design to the synthesis to application, so we will be able to tackle these fundamental challenges like far more quickly and more powerfully. So as a summary, hopefully, today I have been showing you my dream of creating a tunable crystalline materials and how, AI can actually revolutionize this process. So I would like to express my deepest gratitude to my advisors, Professor Omar Yagi and Professor Klaus Jensen, for their invaluable guidance. I would also like to thank the Dream Chemistry Award Committee for giving me this wonderful platform to share my ideas and aspirations. And finally, I would like to thank all of you for your attention and being here today. I'm really looking forward to the questions. Thank you.
Thank you. Questions, comments?
Thank you very much. It's a beautiful dream again, another one. And I have a question about, is it possible to include as an objective to calculate the fatigue resistance of these materials? Because sometimes upon several desorption cycles, they become, they fall apart simply and deteriorate. Do you think it will be possible to do that, these kind of calculations for AI?
That's a great question. So if just to clarify, I understand your question. So you are talking about resistance, or you are talking about stability?
Yes.
Yeah, okay. Yeah, in this case, so definitely I think, in the future there will be we'll be able to calculate the stability for this material. So there will be two ways. So one is, more about the computational side, which, we can think of there, like kind of in terms of energy, like, how thermodynamically stable they can be, if it consists of the, metals and the linker binding that just make them unstable. So AI will be able to, incorporate these, numbers from, like, let's say DFT calculations as evaluation criteria. And on the other side, we can also think about their experimental, measurement outcome. So one thing, I know, some other people actually are working on, which is excellent work, is that they try to extract the data from the TGA, the thermographic analysis data. So for each moth, there will be a decomposition, temperature. And if this temperature is low, that means this moth is less, stable, especially in terms of operation condition. So they use this number to, to make a judgment in terms of whether or not it's stable or not. And it's also some kind of, related to the long-term stability, because, if you have a moth that can operate at a very high temperature, and it has a very high decomposition temperature, so it will be more stable in the long run. So the short answer is yes, definitely we can include that. And that's really a very good point, for the real world application considerations.
Yeah, thank you.
Yeah, maybe a follow-up. So this is a game of catch and release, right? And you talked about the catch and optimize the catch. Does it also make sense to optimize the release conditions?
Great question. Yeah. That's actually another very good point. So in some case, some of the moth has the in this case, so we are mostly in the slide, mostly talking about the storage, for the gas molecules. And in terms of the release, so I think it varies from, moth to moth. And definitely, we can try to think about, how to incorporate the experimental data, especially in terms of measurement, about how to optimize their, release capabilities. I guess this will be the next level after the capture, because this requires, other data, this kind of release, data, and also, some kind of experiment data in this case. And a lot of time, is missing in the literature. But definitely this is something that is possible. But I guess it will be even the next level. Yeah.
Thank you.
So thank you very much for the talk. I have in front of me a paper from Nature Communication 2024, and the first sentence from the abstract is: ChatMoth is an artificial intelligence AI system that is built to predict and generate metal organic frameworks. And then following sentences: ChatMoth shows high accuracy rates of 96.9 for searching and 95.7 for predicting moths. What's different, what is special, and what's unique about the alpha moth versus already published dream of others? Thank you.
Okay, yeah, that's also a great question. So I also saw the great paper from Professor King. So I think here the major difference I will say is that in his case, so he's mostly interested in, trying to enumerate all the possibilities of, trying to, based on my understanding, trying to, assemble moth, which means that you need to give a specific, building blocks. And then, so in this case, it will try to, assign you one of the topology. But in this case, so what I'm trying to see is that I want to use all these experimental data to, predict, the eventual topology and how they eventually assemble into the 3D structure. And in this case, the difference will be whether or not you know which topology you will end up with. So it's like, the folding of proteins. So I think, currently our approach is that, we just try to enumerate all the possible, topology. There can be, 20 or 30 of them. And, ideally, you can try to just fit all of them into your, prediction, which will be more computational intensive. And the other consideration is that a lot of these topologies, so just as I say, synthetically, they really depends on the condition you choose. So even though you slightly vary like a metal or like a linker, so sometimes you will end up with an entirely different topology, or sometimes you use a different solvent, just very minor changes, a lot of different stuff. So my dream will be more focused on trying to make this to be more experimentally sound, or more like synthetically feasible by relying more on the experimental data rather than the de novo design, or the like trying to enumerate the possibilities. So I guess that's the main difference. But I need to acknowledge that currently there is a lot of great work also in other approach, and eventually we are all just aiming for the same biggest goal, which is can we try to predict it accurately, and actually also trying to first predict and then to make it synthesizable in the lab, because that's the more important part.
Are there any questions or comments? If not, let's thank Z-Link once again.
All right, yeah. Thank you very much, and it's been a great morning so far, and it's a true privilege and a pleasure for me to now have the opportunity to really present my dream and ambition going forward with my independent research. And this dream and ambition really revolves establishing a data-driven platform to unleash probably nature's strongest design force, Darwinian evolution, to design and synthesize sustainable peptide-based materials. And I'm very passionate about these peptide materials because I strongly believe that they can play a pivotal role in one of our society's biggest challenges. Because even to this day, over 85% of our materials and materials chemistry that we do is based on petrochemical feedstocks and fossil fuel energy. Yet we know that because of the dwindling supplies and the climate issues, we know that over the course of our lifetime, or at least for the younger ones in the audience here, we will really have to shift away that old-fashioned materials chemistry towards a new bio-based and biodegradable materials science. And this old-fashioned materials chemistry is problematic for a number of reasons. As I mentioned, first off, during the mining as well as the production, the use and the wasting of these materials, we're emitting huge amounts of carbon dioxide, so not just by burning it, but also by using the materials. And we're only expected to make up for these emissions to a limited extent by increased use of green energy or more efficient production processes. And actually the prospects of recycling or CO2 fixation into new types of materials with the current technologies are looking pretty grim. So that leaves us with a huge gap of potential CO2 emissions that will pollute our atmosphere and turn this planet in a potentially unlivable place for future generations. And it's not just the CO2 emissions that's problematic of our materials chemistry. It's also the materials themselves. So as a Dutch person that likes to go on sort of extended bike rides on our flat countryside, I deeply care about products such as this chain loop here. So these products you can use to put on your bike and make sure that your chain goes running smoothly so you can keep on cycling personal records. However, this particular type of chain loop here is actually sort of based on chemistry developed in the 50s, 60s, and 70s. And as you can see, it's containing PTFE, which works very well, but that also means that when you put this stuff on your chain and you go onto the countryside or you go into the forest for a nice weekend ride, you're basically shedding these Teflon particles all over the place. And these particles will persist there for decades to come, disrupt many biological processes, and really present a problem for future generations. And this is just one example of these sort of materials chemistry that we use in everyday products, but are really released into the environment and polluting our environment. You can, for instance, also think about microplastics in shampoo, or lots of paintings, coats, or stabilizing agents of emulsions and dispersions, for instance. So this really goes to show that we really have to work hard towards replacing this old-fashioned materials chemistry with new bio-based and biodegradable alternatives. The problem, however, is that we like these types of materials because over decades of research on these petrochemical-based chemistry, we know very well how to get excellent performance out of these materials at an affordable price. For many of the bio-based alternatives, which are typically a different class of molecules, we just oftentimes simply don't have a clue how to get similar performance out of a different class of molecules. However, it's not that it is impossible to get good performance out of bio-based materials, because basically every day when you go into the lab and you look at your colleagues, you can witness the great beauty and diversity of the materials that biology can make out of proteins and peptides, right? Ranging, for instance, from keratins, which can be found in hairs or in nails, or collagen, which skin, or even softer elastins, or maybe when in the morning you go for a morning walk and you can witness the morning dew on spider webs, you can really witness the strength and the elasticity of these bio-based materials. Or maybe when you're sick, you need some peptide-based antibiotics to make you fit again. And to me, as a materials chemist, this really goes to show that nature, over the course of millions of years, was able to use this super strong design force, Darwinian evolution, to get this huge breadth of material properties out of peptide and protein-based materials. And to do that, it just needs a limited set of 20 amino acid building blocks. And so to me, as a materials chemist, it really goes to show that as a materials chemistry community, we should not feel intimidated by the limitations of these 20 amino acid building blocks, but really embrace that potential of Darwinian evolution to generate the materials that we need for our own technological needs. Now, I can imagine you're thinking, this is all nice and clear and everything, but I do know that the Nobel Prize this year was awarded for protein design and structure prediction, yet now I stand here telling you that we sort of cannot make materials out of these amino acids. How does that work? And so I deeply agree with the Nobel Prize's recognition of the importance of the protein structure design and prediction. And it was actually in 2021 that the Google DeepMind team and the Baker team released their AlphaFold 2 and Rosetta Fold structure prediction methods. This was actually on my birthday, so I typically don't receive gifts from the scientific community, but I think these two papers were really, really awesome gifts on my birthday, actually. And later work also by Baker and Kim at Stanford also showed that not only can we convert the sequence into a certain protein structure, but we can also design protein sequences that have specific desired protein structures. Now, this is great, and it's been really unleashing an absolute revolution in biology. However, these structure prediction design methods are kind of special because they're trained on huge amounts of structural data in the protein data bank. And this data is basically data on very large, hundreds of amino acid long protein sequences that form a specific structure in an aqueous solvent. As a result, we've become very good in predicting and designing protein structures in these aqueous solvents. However, when we want to study molecules and materials, protein-based materials that contain, for instance, modified amino acids such as ornithine, phosphocyrene, or many other modified amino acids, or we want to study the dynamics of proteins and peptides, for instance, in the case of studying protein folding or studying intrinsically disordered proteins, which is very important for neurodegenerative diseases, these current design methods already start to have some of their difficulties. And if we go really down to the area of material science, for instance, if we think about replacing that Teflon-based chain loop with bio-based alternatives based on peptides and proteins, we're typically very much interested in the properties of our materials at long length and time scales. And for instance, for these formulations, very much interested as well in what are the properties of our materials, our molecules in dynamic complex media, for instance, with additional salts, surfactants, or maybe a bit of organic solvents. And it's really in these areas that these current structure prediction methods have no clue at all or have no use at all in sort of predicting the properties. In fact, for many of our sort of consumer type products that we are actually using today, these large protein structures might not be that great to start off with in the beginning, because these proteins are actually, they can be difficult to produce, expensive to produce, difficult to handle as well. They might not be so resistant against certain environmental perturbations such as pH or temperature, for instance. So in this regime, peptides that are of intermediate length up to about 10 to 20 amino acids present an ideal compromise where there's a bio-based material that has a high degree of functionality from the diversity, the chemical diversity of the peptide molecule, as well as an ease of handling and an ease of production where we can afford these molecules relatively cheap. However, the properties of these peptides of intermediate length are difficult to predict because they are not determined by secondary structure elements such as alpha helices or beta sheets, but rather more by sort of intermolecular dynamics that are more reminiscent of small molecules. And as a result of this different sort of origin of their molecular properties, we cannot actually really predict much of their structures, or much of their properties. And this really goes down to really basic material properties. So to get a little bit of an idea of here as a sort of an audience, we can have some sense of the properties of simple dipeptides. Let's see if we can get an idea of the solubility of a simple dipeptide. So I'm giving you a way that alanine is soluble in water up to about 89 grams per liter. Glutamine is soluble about 36 grams per liter. So if we chain these molecules together, so give me some hands, who of you thinks that this dipeptide now all of a sudden becomes about as soluble as the sugars? Right, so no one. Who thinks that this molecule all of a sudden starts to basically become as soluble as a rock, so hardly soluble at all? So some people, who of you think that this is then a trick question? No, everyone should. All right. No, so it turns out actually that when you make the dipeptide, the solubility of alanine glutamine shoots up to a whopping more than half a kilo per liter. In fact, this molecule is so soluble that it's actually used as a prodrug for medical infusions for people that need high amounts of glutamine. This, however, also goes to show that the basic physicochemical properties of larger peptides are really highly nonlinear combinations of the amino acid building blocks. And these highly nonlinear combinations makes predicting the properties of these peptides very, very difficult. And in fact, many of these properties are actually not even known in the publicly available databases. So solubility, for instance, of dipeptides, there are some, but it doesn't go into the hundreds. If you go to tripeptides, already really is sort of anecdotal, and larger peptides, we basically cannot find any solubility data in the databases. So as a result, data-driven prediction of much of peptide science is currently still impossible. Predicting, for instance, what the influence of pH salts solvent or temperature is on basic peptide properties, such as their tendency to aggregate, is currently not possible. Or, for instance, predicting how a certain peptide, what sort of the basic physicochemical properties, such as solubility, interfacial tension, or other basic fundamental simple properties are of a given peptide sequence is also still very much an open question. And so to give you a little bit of an idea of the state of the art of peptide materials in 2024, which is, I remind you, the age of ChatGPT and shooting stuff to Mars, I here present you one of the latest materials published this year in 2024 in the journal Nature. So as you can see, it's just a very simple tripeptide of tyrosine. And actually the authors were able to show that with a bit of processing, they can make this material into a hard, fully transparent, bio-based and biodegradable material that can, for instance, be used as lenses. To me, that also goes to show that there's a huge wealth of beautiful materials out there that remain to be discovered, if only we can navigate that peptide sequence space a bit more efficiently. And to do this, data is really key. And this data we are currently lacking. Actually, acquiring the data in the first place is not that trivial, because already at short peptide lengths, the combinatorial explosion of the possible peptide sequences, all of which can have different material properties, makes that measuring that sequence space is actually very difficult. And so, in order to realize this data-driven prediction or this data-driven design of peptide materials, we really have to do better. And this now brings me to my dream, which is the realization of an AlphaFold-like breakthrough that can enable data-driven design of peptide materials that enable further optimization using nature's power of Darwinian evolution to fully design the materials that we require for technological purposes. Realizing this dream entails the construction of a foundation model that can efficiently map the chemical structure of the peptide to the intrinsic physicochemical properties of that molecule, which then enables us to efficiently optimize initial candidates for our desired material properties, and which we can then submit to Darwinian evolution through directed evolution to fully optimize the materials for the applications we desire. Constructing this foundation model first means that we have to start building an active learning workflow to generate the high volumes of data that we need to train such a model. And so currently in our labs in Nijmegen in the Netherlands, we are establishing such a workflow where we've acquired basically an automated robot platform that can perform solid phase peptide synthesis in a highly parallelized way. And this is now giving us access to hundreds to 1,000 peptides on a weekly basis. And with the advent of cheap liquid handling platforms such as the OpenTrons robots, and we can now also prepare these samples in a highly automated and also importantly highly reproducible manner. And this way the automatically generated samples can be fed into a high throughput analytical workflow that can measure basic physicochemical properties of peptides. And with this high throughput analytical workflow, we can then start training machine learning algorithms to predict the properties of new and unseen peptides. And with Bayesian optimization methods, which have over the last couple of years really been revolutionizing the chemistry, the chemical field, we will then select new peptide sequences that after experimental validation with the platforms here, will maximally improve the predictive capabilities of our model. Now for this high throughput analytical workflow, I specifically propose not to focus on in-depth characterizations of a select number of peptides, but rather to just make clever use of basic instruments that probably here you also have standing around in the lab. For instance, using just standard plate reader equipment or nephelometry equipment, one can easily measure solubility of peptides by looking at sort of scattered light of non-dissolved particles. Or for instance, we can measure electronic properties of peptides by measuring absorbance of fluorescence spectra. Or we use basic HPLC equipment to measure concentration of peptides, either in octanol or water phase, to determine actually the log P. And there's a couple of other sort of basic physical-chemical analytical platforms that we are currently developing, and I'm happy to discuss these further in the discussion section. And I strongly believe that these basic physical-chemical properties are very important to measure, because ultimately these basic, simple, fundamental properties are what will ultimately drive the suitability of your material for more technologically advanced applications. And so with these basic high throughput methods that are actually very affordable to many of us, we can then generate data that we can use to start doing machine learning. We're particularly thinking about using Gaussian processes-based machine learning models, because these Gaussian processes machine learning models already enable machine learning in the low data regime. Moreover, these Gaussian processes can also in principle model any structure property relationship, and besides giving you a value for a certain property of the peptide molecule, let's say its solubility, it also intrinsically gives an uncertainty on that prediction. And this uncertainty is massively helpful, because we can query the model and ask for which peptide sequences it actually doesn't have a single clue what it should predict. And of course, these are the peptide sequences that are the most informative for experimental validation with our experimental platform. And upon experimentally determining these values, we can then retrain the model in such a way that it maximally improves its predictive capabilities. And by iterating through this loop, we will then gradually improve the predictive capabilities of our model and ultimately learn to sort of predict peptide material properties. Now, where to start in this rather sort of large data collection campaign is very much a very important question. And to address this question, I propose to use a curriculum learning approach. Intuitively, this curriculum learning approach is, I think, very simple. So for instance, for me, my Polish is very bad. So if I would start learning Polish, I probably would first start with, simple books that, I don't know, could start with stuff like Adama Kota, which I believe is before moving on to sort of the more complex stuff, such as Litwo, Ojiczni Moja, right? That's this. So in the case of, well, ultimately we want to get at that stage, right? We want to go towards these types of texts. So for our peptide data collection campaign, that really means that we'll start off with just very simple, very basic peptide properties or peptide molecules, such as, for instance, the tripeptides. So tripeptide sequence space is relatively small, which means that with our automated platforms, we can actually cover a sizable fraction of that sequence space. Initially, our machine learning models will have not seen many peptide molecules, so the uncertainty on the prediction of the machine learning models will be relatively high. But as we iterate through our active learning cycles, the prediction will become more certain. And at some point, we will become able to predict physical-chemical properties of trimers. Now, having graduated from that curriculum grade, let's say, we can move on to more complex problems, such as, for instance, pentapeptide sequence space. Pentapeptide sequence space is considerably larger than trimer sequence space, but because we already have seen many trimer sequences, the model predictions will be comparatively certain. And as such, we will probably need a rather continuous amount of experiments to actually also learn to predict pentapeptide sequences. And in this way, we can then gradually work our way, graduate our way, as if you go through school, through the different lengths of the peptide sequence space, and then ultimately, we will then generate a maximally informative and comprehensive data set on the physical-chemical properties of peptides that will enable us to train that foundation model. And here, we're particularly thinking about training a graph neural network to predict basic physical-chemical properties such as solubility, interfacial tension, and other properties of these peptide molecules. These graph neural networks consider the peptide molecule as a collection of atoms that's connected through sort of chemical bonds. And through the successive layers of this graph neural network, it can condense that chemical information into an abstract numerical representation that can be used to fine-tune on these properties. And by fine-tuning on these properties, this latent embedding adopts a physical-chemically meaningful shape in which physical-chemical or peptide molecules with a certain or peptide molecules are clustered according to their intrinsic physicochemical properties. Now, recent work has also shown that not just using chemical connectivity, but also sort of conformational through space interactions can additionally improve the performance of such graph neural networks. And for instance, by making use of machine learned potentials, we can now efficiently map conformational space of these peptide molecules. And in this way also sort of infuse conformational dynamics into our connectivity matrices that we feed into the graph neural network. So as such, we can then ultimately learn a maximally meaningful physicochemical space in which the peptide molecules sort of live. And as such, we obtain an efficient mapping of the chemical structure to the physicochemical sort of meaning of these molecules. Such physicochemical or such efficient mapping then will allow us, from these basic physicochemical properties, to start doing transfer learning, where we can adopt our active learning approaches that we developed in the early phases of the project to optimize sort of examples from the literature towards new properties such as conductivity, viscous elasticity, and let's say toxicity. However, as our foundation model is trained on just proteinogenic amino acids, it will do very good in probably predicting these molecules, but for many applications, let's say for instance conductivity, the proteinogenic amino acids might not be that great, or might not be the best candidate to fit our technological needs. And so for this, I propose to sort of now ask Mother Nature just to do that optimization for us, rather than us having to go to the whole trouble of making another foundation model. And so to do so, we will then have to transfer the candidate molecules that are suggested and optimized with the help of our foundation model into a living system to which we can apply an evolutionary pressure, and then also have that system able to expand the chemical diversity of our peptide molecules. And in this respect, I'm particularly excited about two different types of biochemistry that can diversify the peptide chemical structures. The first is the biosynthesis through so-called non-ribosomal peptide synthetases. And you can think of these molecules as sort of beads on a string, and where the small beads here install incoming amino acid building blocks into the growing polypeptide chain. And there are certain amino acid positions here, and these positions that sort of specify which amino acid building block will be incorporated. And these larger pink or dark blue domains then further modify the chemistry of these amino acids. And so to give you a little bit of an idea of what type of chemistries actually are accessible with these molecules, you can here see the biosynthetic, or the final stages of the biosynthesis of bleomycin, which is a peptide-based natural product, can actually be used as an antibiotic. And we see that in the final stages, this blue adenylation domain here introduces a cysteine molecule into the growing polypeptide chain. And this orange cyclization domain then converts this cysteine molecule into this thiazole ring here. And in the final module of the biosynthesis, a similar module actually introduces another thiazole to finally yield this bis thiazole moiety here. And now with advances in engineering these assembly lines, it's now, by us and others, it's now becoming very much possible to sort of generate larger, fully synthetic assembly lines that can really yield molecules that are looking like this. So it's important to realize that these molecules are fully bio-based, derived from peptides, but would actually, I believe, not be so out of place in papers on organic electronics, or so organic photovoltaics and stuff like that. A second approach to transfer these systems into biological systems, or transfer these peptide molecules into biological systems, is biosynthesis through ribosomally synthesized and post-translationally modified peptide synthetases. Potentially, these approaches is a bit more amenable for directed evolution, but engineering approaches here are more in the earlier phases. Now, this RIP biosynthesis, or the RIP product, is actually encoded on the genome, and within the same operon, we have additional modifying domains that modify and expand the chemical complexity of these molecules. And so, upon translation of these operons, the actual precursor towards the complex peptide is formed, and the modifying domains recognize that the leader part of the peptide and modify the core peptide, which will be the ultimate material. And so again, to give you a little bit of an example, we can see, for instance, here in the biosynthesis of the muscaride A natural product, we can nicely see how the modifying enzymes, here for instance the heterocyclase and the oxidase, convert this genomically encoded bis-threonine molecule into these bis-oxazole moiety here, where again we can easily imagine this would be useful in, for instance, organic electronics applications. And so as such, we started to sort of realize a scheme where we have our foundation model that can propose and optimize an initial candidate And we can encode this peptide molecule into a living system. And with the use of directed evolution, we can apply evolutionary pressures to sort of fully optimize these molecules into broader regions of chemical space, so that we have the optimal candidate to finally sort of start thinking or start realizing, really replacing this old-fashioned sort of chain loop and all the other petrochemically based materials that we're using in, for instance, our consumer products, with sort of bio-based and green biodegradable alternatives to make sure that also this planet and the forests and the woods and everything remain a livable place for future generations to come. With this, it leaves me nothing else to thank my team, specifically Justin, Andre, Wojciech, Idem, and Pam, who've been really working in Nijmegen a joy over the last year, starting up the research group. The other colleagues in Nijmegen, Big Chemistry, for the funding, and then finally you for your attention. Thank you.
So how can you imagine producing 100 million tons of this type of materials?
That's a
right,
good
because
question.
you want to replace them. So 100 million tons is just a very small estimate of how much you really need of the stuff. So
Yeah.
how would you produce it?
So we can discuss about the applicability of this method for really bulk commodity plastics like polyethylene. On the other hand, we are producing through fermentation, right, biological production, on huge scales, bioethanol, for instance, and companies like Corbion have become basically the large bourses. I think so, yeah. The great thing about this bio approaches is that in principle they're scalable, because once you basically have your organism that produces it, it just multiplies by itself.
So, okay, one of the major problems with the short peptides is the actually susceptibility to the proteolytic degradation. And I'm not talking about the human proteases here or peptidases, I'm talking about the bacteria proteases.
Yeah.
Where actually you can build or you can, design perfect molecule, and then this molecule can be immediately digested by the bacteria. So on one side, you have the advantage because it's the biodegradable material.
Yeah.
But on the other side, how would you check actually all of these compounds for the stability, the proteolytic stability in all possible conditions?
So I think this is where this directed evolution approach comes in very nicely, because basically, so these peptide synthesis or this peptide biosynthesis pathways, in a way, are very similar to what we try to do. Just typically, the biosynthesis of these peptides is based for chemical warfare for bacteria against each other. So biology basically solved that, or has to deal with that problem too, and in a way has designed, well, enzymatic pathways that introduce chemistry into these products that can actually circumvent this degradation.
Yeah, sometimes it actually can enhance. And I'm working actually on very similar projects. And actually, if you modify the side chains of amino acids, sometimes you can get super fast degradation by proteases. So yeah, then you have actually two sides of the same actually.
Yeah, exactly. So it is known, for instance, for these RIP, the last example, the RIP biosynthesis that I showed you before. There are also, for instance, epimerases that are known that convert these L amino acids into D amino acids. And for these, it's, for instance, typically known that the proteolytic stability actually increases. Or we can, for instance, attach lipid tails, for instance, which in MedChem also have a tendency to increase half-life and proteolytic stability. But
So
we have to, it's one of the, it will be one of the targets that we have to optimize for.
What about using unnatural amino acids?
Also possible. In a way, it depends a bit on how we would do it. The RIP biosynthesis, for instance, we could think about using genetic code expanded organisms to indeed directly already encode those in the genome. If we go the NRPS, so the bigger enzymatic route, there are similar enzymatic domains that can also introduce these unnatural amino acids. I think ultimately that's the way you have to go, because nature knows very well how to degrade proteinogenic amino acids, but sort of unnatural amino acids a bit less. Yeah, fully agree.
Thank you.
Okay, I like your graduation program where you train on the short peptides hard, and then it's easy for the longer ones. But an obvious devil's advocate question is that many of the properties you seek for will be depending on the secondary structure of the peptides, and secondary structure only builds at a certain length. That's definitely longer than your graduation program. So what do you do about that?
Yeah, so basically you start seeing secondary structures. You need some size of amino acids, so some length of the peptide. I think already, also if you look at sort of the current examples out there, where for instance, just from a simple tripeptide, you can already make a mechanically rigid, transparent gel that actually could, function as a glass. I think we can already replace many of these sort of old-fashioned materials in short peptide sequence space, and where these secondary structures probably are only a minor concern. I mean, you need, what, 3.14 or something amino acids for an alpha helix, but can you really make a tetrapeptide alpha helix? I don't think so. You probably need some additional stabilizing forces, because entropy and enthalpy for such small molecules play out differently.
Exactly. So if you would like to target properties that are dependent on the secondary structure, do you have like an alternative strategy than this learning from the short ones, or?
Well, in a way, I mean, if we sort of use our active learning scheme, or also the directed evolution, we don't really care about the structure. We ultimately care about the property. So if it turns out that we find a molecule that has a certain secondary structure, that's great. But ultimately, we are interested in properties, right? I mean,
but chemical intuition tells us, right, that the secondary structure is very often connected with the property, particularly for enzymes and like this. So
and we can also
maybe it's not a good idea to throw this knowledge completely away, right? But rather somehow maybe try to embed it in the learning. So that's why I'm asking whether there's any way you can see how to embed this.
So I can see sort of in the longer run where one would also incorporate sort of the approaches that Zach was also showing, where you use sort of large language models to also start doing hypothesis generation. I think now over the last sort of year or so, we're starting to see the first examples of how people are able to do that. At the moment, that's still in the very early phases, I would say. But I think I can imagine sort of over the next decade, this type of hypothesis generation and, well, physics-informed or chemistry-informed AI will really take off, and we'll have to take that into account and sort of infuse that in our design processes too. Yeah.
Thanks.
Thank you for a very nice talk. I have a question from a point of view of someone who is completely out of this field.
Mm-hmm.
And I have this very naive question. So when I'm thinking a protein-rich material, I'm thinking, for example, a piece of meat in my fridge. And we all know what happens if we leave it for longer than a few days.
Yeah.
So
Starts
can
to get
those
smelly.
materials really, do you think those materials can be really made stable enough to fulfill the functions, and how to achieve that? And are you aware of any materials that are already out there that are really, that are peptide based and are used for technological purposes somehow? So I really don't know. So it's an honest question.
So to address the first part of your question, that basically also is very related to the question of your neighbor. How can you sort of tune that biodegradability? So I think the way to go there is to basically use these such types of approaches where we can use directed evolution, for instance, to diversify and expand the chemical scope. And you can think of sort of directed evolution approaches or assays where you make an assay that's based on the property of that molecule, but you also introduce peptidases, for instance, that would sort of chew away on non-stable peptides. So I think in this way you could really also circumvent that problem and generate stable materials that are still based on natural resources, but have increased proteolytic stability. Now, for the actual examples of materials, this is also a good question. I mean, so of course there are some protein-based materials out there already. I mean, stuff like gelatin and stuff like that. It's also in a way protein-based or peptide-based. The material properties are maybe not spectacular, and in fact, so it turns out that sort of this bio-based material design is also interesting to many companies, because especially in Europe now we're having this Green New Deal and we need to phase out, for instance, all the Teflon-based stuff. And when we're talking also to companies like, hey, what type of materials are you interested in? Basically, it turns out that there's not that many of these peptide-based materials on the market yet, but there's really a strong interest in sort of making these materials comparable to our petrochemical materials that we are currently using.
Thank you.
Very interesting talk. My question is also about the curriculum learning section. One question I had is that you mentioned that the downstream properties are not always very easily predictable from the constituent parts. So do you think that there's like a threshold number of amino acids where, you know, like knowing, for example, n-1, the function of those a length n-1 is no longer that helpful for length n because there might be some novel interactions? And related to that, do you think that instead of doing it by number of amino acids, there's some other way you can break it up into curricula? Like I'm thinking in terms of language, you don't really learn all of the 3-word sentences before you move on to 4-word sentences. There's maybe some other structure that might be a better kind of divide for the curricula. Thank you.
Yeah, so basically to relate to your first question, what's sort of the scale that we have to think of, right? So there's hardly any literature basically on active learning and this type of curriculum learning for peptide materials, so we'll have to see. However, for other areas of chemistry where people actually are using this active learning already more frequently, typically you see that with really fractions of the total space, so really maybe 1% or sometimes even less, you can already train good models that are actually able to predict the properties of your system in a good manner. So really orders of magnitude less than covering the full sequence space. To address your question of is the trimer, pentamer, heptamer, is that really the best approach? Also I think goes a little bit to Pavel's question, what about secondary structure? Because in a way, as we grow in complexity, at some point we might start getting certain patterns in certain properties. If there are indeed these patterns, I'm very optimistic that the machine learning will actually pick up on this. And when we sort of query the model, like, hey, for which sequences you actually don't know what, I mean, you give a prediction, but you're very unsure of that prediction. Probably for those larger sequences that fall into such categories, the model will say, well, it's probably going to be this, and I'm really quite sure because, you know, for the shorter peptides, we start to see a pattern.
Just to relate a little bit to that, when you look at normal polymers from in all of our plastic products, a lot of the changes also in polymer properties comes from things like cross-linking and such things. How would you predict that using models of these?
Yeah,
I mean, you can't predict the models of a monomer of a cross-linked polymer by looking at its monomers, right?
Yeah, that's a good point. So maybe also, I mean, so there's also the question of, can you do this on megaton scale, replacing, let's say, polyethylene? So you're also thinking about, for instance, the properties of polysaccharides, right? I mean, there's so many polysaccharides that are basically based out of the same, pretty much the same molecule, all with wildly different properties. I think at the moment we simply don't know. Basically, what you see is that, for instance, this whole field of AI for chemistry in polymer science is really at the early stages, precisely because of this reason. How do you deal with things like crosslinks? How do you deal with sort of gradients in your polymers, etc.? I think we're still waiting for those types of material questions and material applications for further advances in the field.
So more than 20 years ago, there was a structure property model that is used in industrial companies like Cosmo RS, which is exactly, you give a structure and you get the property like solubility or something that. They are better or worse. But how the outcomes of these models compare, or how you would imagine that they will compare to the to your scheme that you propose?
Yeah, so in a way, these I'm vaguely familiar with Cosmo RS, not too much, but I know of its existence. What I can say is that, for instance, for sort of your basic predictors that you would use for machine learning on peptides, sort of the standard ones that you could calculate are really kind of just linear combinations of the amino acid properties. And basically, we've shown here with the audience that it's really difficult actually to predict, basic properties such as solubility by these linear combinations. So by really looking at the molecules as individual molecules, as a specific class of molecules with highly nonlinear interactions, I think we could probably do better than Cosmo RS by maximally or training the model on maximally.
I think it would be interesting to combine the two words, because Cosmo RS has structures inside, you can combine the structure information and maybe predict some new things by combination with the experimental findings.
Yeah. No, I think ultimately this is really the way forward, where not just doing the calculations, but really basing the calculation, or having calculation-informed experiments and experiment-informed calculations. Yeah.
Thank you, Matthijs.
All right, thank you.
First of all, thank you very much for the opportunity to dream. So to Robert and to Pavel and to the hospitality of the Polish Academy, it's been quite some, a really fun day for science and really as a celebration. So as Robert mentioned, that there's not that many awards for dreaming. So when I was thinking about my dream, I really dreamed hard. So I apologize if I had to break some barriers in my field and to sort of like started looking into different directions. But I wanted to really make it feel like a dream. Because as a solid state chemist, we don't get to dream much. And also, my dream begins in this image of a textbook when I was a budding solid state chemist trying to learn about the physics and chemistry of semiconductor materials, but also at the same time, naively dreaming about trying to harness the chemistry that we have to revolutionize the materials building blocks that we use for modern transistors. And naively I said because obviously it's not that easy, and this image is quite inspiring in such a way that it did not only pave way towards the modern computing generation, but also at the same time, it was one of those few demonstrations back in Bell Labs that enabled us humans to manipulate matter and to control how electrons would move in a semiconducting material with just an electric field. As a solid-state chemist, this is quite surreal to talk about my dream in this regard and thinking about transistors and integrated circuits because of the Sokolsky method, which was developed by John Sokolsky, which really enabled a lot of solid-state chemistry, but also at the same time revolutionized the way how we can think about high-quality solid-state crystals that we can turn into wafers. So fast forward 70 years, of course, things have changed in decades. 70 years of physics, chemistry, and materials engineering have led us to the modern transistor that we see in our cell phones, in our computers. And now we see from that one transistor the size of my thumb, decades after, we have billions of these transistors. And to just put something into perspective, the first lunar module that sent the first humans to the moon only had 20,000 of these transistors. So we've really gotten far, and we need more. We've seen a lot of very interesting talks today that talks about how we can harness artificial intelligence, sometimes augmented reality and large data sets to move the quality of life forward, and we would need more computing power for that. Of course, this is not going to be an engineering talk, so I won't talk about different architectures to optimize this process, but the demands of next generation functional devices, not just the integrated circuit, really is approaching the atomic limit, which is a problem because for 70 years, we have been using silicon nanowires as the basis for these integrated circuits. So an alternative approach is to start thinking about materials that approach the atomic scale thicknesses. And one of the beauty of solid-state chemistry is that we can think about solid-state materials which are low-dimensional and are held by weak van der Waals interactions like graphene. These weak interactions that we see in graphite enable us to peel off these materials in the fancy process called micromechanical exfoliation using a Scotch tape to access sheets of graphene from that bulk material with good atomic precision and with less than a nanometer in thickness. These are perhaps the thinnest materials that we can see and we can handle. And the precision also enables us to think about the material's properties and model them and see that one-to-one in the experiment. So from there, chemists jumped in and developed a library of these materials with different properties. The physicists and engineers jumped in as well and started developing methods that we can stack these quite literally one layer on top of another and accessing complex functionalities based off of these monolayers. Something that is quite inspiring is that Taiwan Semiconductor Manufacturing Company and Intel started integrating these materials, really highlighting for the first time that we're moving from silicon and we're starting to poke into this new direction of accessing new materials that we can fit into our modern transistors, processors, and integrated circuits. However, when we start looking into this process, we encounter yet again another problem. So when we shrink silicon down close to the atomic scale, silicon becomes sand. It becomes oxidizes. It doesn't behave as silicon anymore. When we access one atom layer thick or several atom thick layers, we take care of one dimension, but when we start shrinking these into the size of the smallest limit, then we see these dangling bonds or radicals or unpaired electrons that impart non-intrinsic edge state chemistry in these materials, turning these into other materials as well. So where do we then go beyond this two-dimensional limit? And in these, I quote two big leaders in Silicon Valley, really trying to ask how small can materials building blocks be, or how complex can materials building blocks be in terms of really developing materials that would enable us to advance the integrated circuit and the components that comprise that circuit. So in a way, I'll pose this question: is there a pathway to access precision in solid-state materials with such complexity that will take the current way of modalities with which we do computation or processing in these integrated circuits in a slightly different way? And with that, I'll pivot a little bit. As a solid-state chemist, I've always been envious of my organic and chem bio colleagues who get to work with biomacromolecules, because these materials are both small and complex. And we've seen, Matthias's talk today in terms of the potential of these materials and the potential of sequence-defined structures and properties in these structures. So if we look into nature and we look into how nature runs its own integrated circuits, biology happens because we have long-range, directional, and high-fidelity and error-free transport of charges and ions. Of course, I won't go into the biology, but since I'm a solid-state chemist, I'm almost a physicist. We think about this in the physics perspective. So why did nature opt for chiral materials and helical materials instead of the metals that are also available in nature? Well, it turns out we can look at this in a simple model, right? If you have a helical material, apply a current as if it is a solenoid in the atomic scale, you have a chiral electric field and a velocity, V cross E, you get a magnetic field that it turns out somehow enables us to use these chiral helical structures as a spin filter to select either spin up or spin down electrons. So this is quite bizarre for me, because now we can use biomacromolecules for quantum computing. And this is not a concept. This has been demonstrated and has been shown in many different examples. I'm showing one way to explain it. There's so many ways to look into this problem now. But that enables spin-selective transport, the absorption and emission of circularly polarized light, and even enantiomer separations in the surfaces of these materials. Beyond that chiral-induced properties, these are beautiful building blocks. These are very deep chemical phase spaces, which we can think about in terms of the sequence of the canonical and non-canonical amino acids that form the secondary structure, that then would fold into tertiary structures, and in some cases can even form these complex aggregation of quaternary structures. So this is a very nice, deep, complex, and small building block. And I don't want to reiterate again the power of de novo design and artificial intelligence and machine learning approaches to start developing, this loop of figuring out from big data, right, several sequences and looking into the analysis and analyzing these patterns to come up with a structure. But in solid-state chemistry, if you open up a solid-state chemistry textbook, we don't have that many rules that organic chemists have. So we will be stuck in this region, really just thinking about even that basic fundamental chemistry that would endow us the opportunity to look at sequences. But this is a dream. So this is going to be a big dream that might not even be 20 years, maybe 50 years ahead, but I'm showing you a possible approach that we can maybe use. So my dream chemistry really relates onto creating complex, long-range ordered, conducting inorganic polymer-like structures that would then assemble or fold into complex structures imparting new axes for conduction or emission of photons and whatnot, really trying to bridge this gap between modern inorganic integrated circuits and the complexity of sequence-dependent structure from biomacromolecular systems. And when I wrote this, and this is before the Nobel Prize came out, this is also a dream for solid-state chemistry. Can we put rational design and rational chemistry in terms of, in the context of AI and really, the way how Rosettefold and AlphaFold figured out these things, can we get some rationality out of these, and can we use that to move the field forward? Because as many of you have read in several papers, in solid-state chemistry, you put elements together, you heat it up, you cool it down, and then hope that something forms. So we're trying to change that. Okay, of course, if I told this to myself as a solid-state chemist 5 years ago, I would never believe myself that this dream would even be possible. Because this dream is only possible if we can work with a materials platform, an inorganic materials platform that resembles polymer-like structures. And again, if you open your chemistry textbook, you won't find any solid-state material that is defined the same way. So is there even a rational synthetic pathway to access these materials in the lab? As a solid-state chemist, we think about stoichiometries and composition and structure relationships. So if we take the two-dimensional structure of elemental bismuth, can we use chemistry as a stoichiometric scissor to cut up one-dimensional chains? It turns out we don't have to reinvent the wheel. We just need to look at binary phase diagrams. At least for us, this is how we found these phases. If you look at the binary phase diagram of bismuth and iodine, one-to-one bismuth iodine leads into this nice structure that still resembles a corrugated sheet, but then now iodine with its one valence electron plugs every bismuth atom. So now we can think of this material as having a one-dimensional structure with weak but anisotropic van der Waals interactions with sub-nanometer thicknesses about 5 to 9 angstroms wide. And because of these weak interchain interactions in this van der Waals space here, we can substitute many atoms, large or small, and that strain that develops is taken care of those space. And when I talk to a room of physicists, I describe these materials as if having a crystalline analog of carbon nanotubes. When I talk to a room of chemists, I describe these as if these are all inorganic, carbon-free polymer structures. If I talk to a room of physical chemists, then you can pick your definition. But what's nice is that these would share some attributes of polymers, such as mechanical robustness. These chains can rearrange, and we can grow centimeter-sized crystals of bismuth iodide, shown here, that we can bend. And these are dense inorganic structures that behave as if they are soft matter in some case. All right, so the dream then will be enabled by opening this deep materials toolbox that would enable us to access the same 1D form factor and create resistors, capacitors, diodes, fiber optics, and so on that we can use as the building blocks for this dream. So in the past 4 years, that is what my lab has been busy about. This was my dream 10 years ago, to really come up with a materials class that would enable us to access helical and chiral all inorganic polymers, semi-metallic and highly conducting one-dimensional wires, materials that emit light in several atoms, chains or a single chain thick, single photon emitters, lasers, and even photonic waveguides. We can harness the growth inside carbon nanotubes to access few to single chains of these materials and access really strong quantum confinement effects, opening a new arsenal for physical properties. And in some cases, we can even use these as materials to drive the assembly of organic monomers. And also with this new lens on, we can look into known structures and also develop and synthesize new structures. And now we have access to several classes of semiconductors, metals, semi-metals, superconductors, magnets, ferroelectrics, and even topological insulators with the same form factor. So with this deep chemical phase space, I would then think about this stage as my rapid eye movement stage. So these glimpses of vivid snapshots that show how this dream can be realized in reality. And first, one of the biggest questions that we ask is, can we make inorganic structures that would resemble helical biomacromolecular structures? It turns out that if you start looking for crystallographic screw axes, a rotation followed by a translation that's parallel to a covalent axis in a crystalline material, you can access helicity in the solid state. And this is quite exciting because if you take out pi stacking interactions, hydrogen bonding interactions, or disulfide bridging, it's really hard to make helical materials. And finding this motif was nice because we can break down this ternary phase into its constituent units. And it turns out that this is quite a deep chemical phase space that is quite modular. So we can access this group 3 site with different group 3 elements and some +3 transition metals. We can substitute different chalcogens. We can substitute different halogenated groups or even organic groups in some few cases into this bond. And it has been predicted that we can even replace this bond or some of these bonds with a group 5 lone pair. So this is a nice Lego toolbox that we can work with. And we have demonstrated several examples, successful examples, that we can change the stoichiometry and the composition of these materials, and we can access unusual helical motifs. So we were able to demonstrate that the gallium-selenium iodide analog crystallizes in a 1D quasi-crystalline motif with a non-repeating helical order called a Bohr-Dicositter helix. if we substitute selenium for sulfur, we have shown that we can access even unusual helical motifs that we don't find in nature. We found a helix with a squircular cross-section. Wikipedia says it's a real word, you can Google it. Photonics folks developed that. But really showing how we can selectively strain or destrain the bonds in a helical material by just changing the composition is quite powerful. And also by changing the composition, of course, we directly alter the electronic and physical states in these materials. We can access different ranges of band gap energies or homolumo gaps in the UV to the visible range, and we gain access to a wide range of curvature and torsion in these materials, so we can form different kinds of helices by just by just compositional substitution. So in a way, I can pull this slinky, stretch it or compress it or twist it. And if you remember the basis for chiral-induced behavior, the curvature and torsion in that chiral field really affects the magnetic field that we can generate in these materials. Of course, we can think of these as dynamic structures as well. We can use stimulus-sensitive measurements, so we can expose these materials to temperature as a stimulus. And as a spring that can expand and contract with strong electron-phonon coupling and phonon anharmonicity, we actually did observe very sensitive changes in the band gap with temperature that rivals the current materials that we use for optical thermometry. And this is in bulk, and we can even exfoliate these materials into several nanowires that we can poke into cells or integrate in different devices. And as promised as well, we can access chiral-induced spin-selective behavior because of the helical axis in this material. And in a hypothetical quasi-2D chiral layer, because of the heavy elements that comprise these structures, we have strong spin-orbit coupling. Really, the main point is to just show that we can have anisotropic spin textures in these materials in a single crystalline platform, that we don't need to create a junction. So you can basically increase the logic functionality of just a single layer of this material, which we can now create in the lab, and we are starting to measure. So with this experimental toolbox, or at least one example of this experimental toolbox, we can then probe, and we can now start asking some of these questions. Can we now alter the interchain sequence or composition of that quasi-tetrahedral building block in such a way that we can introduce a small kink, which is going to be a transistor on its own, or we can develop a block-like structure that's alternating to create pleated sheets or beta sheets, or in some cases, which actually we can see in the lab now, we can program these helices to create super helical coiled coil structures, which really now puts us in a position where we can think about the control of the structure of these materials based off of the compositional sequence. And of course, the dream, of course, will be to harness every computational tool and AI tools that we have to look into program sequences to access different shapes and different complicated structures that we can attach electrodes on using electron beam lithography, and then start looking into axis-dependent transport. So the idea is to start with simple spherical, square, triangular, and even complex octahedral structures, and what sequences will fold into those motifs. So how do we do that synthetically? At least we have some idea on that one. So how do we cue some folding in these inorganic helices? Well, one is we can vary the composition along the chain. By varying the intra-chain atomic composition, we can vary the charge of these materials. We can introduce a region of high or low electron density. We can introduce vacancies, and we can introduce even large or small atoms that will then systematically induce a strain for the system to fold. In some cases, and this is something that we're starting to develop now, is that trial, that group 3 halogen bond is somehow labile, and we can use organolithium chemistry, which we're not very much experts of, but then that gives us an opportunity to use electron withdrawing, electron donating moieties, or bulky or less bulky organic groups to impart helicity and chirality to achiral organic functional groups. But also at the same time, we can start thinking about the sequence and the packing of these structures into these complex superlattice structures, and start interrogating how these structures would fold. But also at the same time, and something that is quite exciting, is that we can now even think about how these folded units can interact with each other, which is something that's not too unheard of. Nanocrystal folks have been thinking about mini bands and intercrystal interactions, so we can also look into those opportunities. Of course, I mentioned that we have some evidences that we will see these in real life. So first, we can grow these materials in the vapor phase, and we can control the stoichiometry, the interchain interactions in not just in these helical materials, but in a wide range of different one-dimensional van der Waals structures to turn on and off those intra or interchain crystallization. In some cases, we find stereochemically active lone pairs that we can turn on and off, and also anisotropies in the composition of how much these materials would need to grow 1D versus 2D. So by designing a pathway where we can control sulfur concentration and antimony sulfide growth, we can actually direct the crystallization of these materials into one-dimensional or quasi-two-dimensional sheets by developing a chemical vapor deposition system that we can selectively control and program the vapor pressures of our precursors before we begin growing. So we have access to wires that are about 5 to 10 nanometers thick and also quasi-two-dimensional sheets from the same building blocks. If we think about the particle-in-a-box problem, this is the particle in a box with different size and dimensions out of the same building blocks, which enables us access to different electronic states. And we actually have experimentally measured that and have shown that we can control that in this paper. Since we have this control over the morphology and the dimensionality with the composition in our synthesis, we now can create 1D, 2D sequences in these structures by altering the concentration of antimony and sulfur that we feed into the system. So you can see a paddle-like structure or a hot dog on a stick structure, and now we're moving towards changing the composition in these phases to access what I mentioned earlier in terms of this composition-dependent growth. Alternatively, we can also use carbon nanotubes as very precise test tubes to grow single chains of these materials and interrogate even atomically resolved structures in these phases, not only showing the drastic quantum confinement effects, but also at the same time having a means to access single chains that are stable in ambient temperatures and ambient conditions, and really using this as a way to control how much we feed into the system. These are melt syntheses that happen through capillary growth as they go inside the carbon nanotube, so we can alter the precursors that we integrate. And of course, one can dream that we can look into chirality transfer from these carbon nanotube into the guest structure, or how the guest structures can sensitize the carbon nanotube properties, and ultimately maybe find a way with which we can control how the carbon nanotubes would then assemble and fold by varying the sequence of the materials that we grow inside of it. And lastly, and this is sort of like going to be the piece de resistance, maybe we can find a way, or we can find some evidences that will enable us to control nanoscale morphology with altering the composition of the chains. And in that, we have our work on niobium trisulfide or metal chalcogenide systems, which are trigonal bicap one-dimensional chains. And we found out that the strong dimerization in the 1D chain arising from Peierls distortion, which is the dimerization to stabilize an unpaired electron in a band, leads to a strong thermodynamic preference that we can turn off mostly the interchain growth and access wires that are about 20 to 50 nanometers thick, but are about 1 centimeter wide, as we have shown here, that won't even fit in a single frame. These materials have high refractive indices like silicon, and that can host mean resonance modes that we can use these for optical wave guiding as well. And really, something that's quite exciting is that when we started looking into sulfur vacancies in the system, we can induce charged regions or strained regions along the chain that we can see periodic or random twisting of the structures. If we start substituting the transition metal with an isovalent transition metal, but with a larger atom like tantalum, at a certain alloying concentration, we can control enough strain in these structures as they grow that the leading end catches with the nucleation point, and we can grow single crystals of rings. So these are a few evidences that show that we can really alter the composition of these materials and will have pronounced influences in how they fold or how they would assemble as we grow them from the vapor phase. So hopefully, with my dream, I was able to convince you that maybe we will see a future comprised of hierarchical integrated circuits comprised of these arrays of folded inorganic structure that would share the sequence-dependent and dynamic properties of biomacromolecules and the delocalized electron properties of inorganic solid-state materials, that we can look into collective and directional interunit states. Because these materials lack center of inversion, we can even start using electric field or magnetic field to influence how they would interact with each other. And of course, we can think of those folded building units as integrated circuits on their own, depending on how they fold, and we can start looking into access-dependent electronic, optical, and magnetic states in these materials. And of course, this is not a pipe dream. We can use cryo-electron diffraction, which we are now using in inorganic structures to interrogate the crystal structures of these folded structures. We can use STM, scanning tunneling microscopy, to interrogate the electrical properties of individual units, and we can even have access to hyperspectral microscopes in my lab that we can look into the optical properties of single nanoscale morphologies. With that, as an educator, one of my biggest dreams is to really enable the dreams of my students, but as you have seen today, it is really my students' hard work and dreams that fueled and inspired my new dreams. So with that, thank you very much, and I'd be happy to answer any questions.
Max, you haven't said much about the electronic properties.
Yes. So did not emphasize the electronic properties because I'm pitching this in the context of one-dimensional structures will fold in different ways. So in a way, these materials actually, depending on what we are intending them to be, for example, for transistor properties, we actually do have semiconducting properties, semiconducting analogs of these materials, which in the context of electron transport are quite interesting if you think about conduction in a helix, because backscattering is forbidden. So actually conductivity restrained in 1D, and with that symmetry protection actually will make really good conductors. If we want to think about magnetic building blocks, we actually do have magnetic analogs as well that we can use for memory storage as well. So in a way, I wanted to show the breadth of the science to really demonstrate that this is a materials class that is new, but is there to last, and there's going to be quite deep. But yeah, and in some cases, we can even make topological insulators which have resistance-free transport out of these materials. So, thanks.
Okay, thanks. So I see the way you aiming to miniaturize, right, and going into smaller and smaller. Yeah. Eventually, at a certain point, it will not be necessarily the size of the molecule, but the fact that you need to redistribute the heat that's during the conduction of the electrons, right?
Yes.
That will be the rate limiting or the size limiting thing. So what can you do with the heat transfer?
Yes, that will be, I think that's a tricky and interesting question, but actually the reason why we were interested in that quasi-periodic 1D structure, that Bohr-de-Cossiter helix, is that if you have an aperiodic lattice, then thermal conductivity management will be a little bit easier, because if we can think about how thermal transport moves in a quasi-crystalline lattice. So that could be something, but one thing that we're trying to interrogate right now is also to now start thinking about heat transfer between the chains as well, which is actually, with how new these structures are, is not too much known. So I think there is an opportunity, and there's ways to mitigate that in context of symmetry protection, but I think it will probably undergo similar treatment in a way how we think about current microelectronics, at least from my perspective, because I work with thermoelectrics as well, and these materials seem to behave very similar to typical semiconductors that we see in thermoelectric materials. Yeah.
Thank you very much. Very nice talk. I would like to ask you about the implication of the quantum confinement effect on some advantages and disadvantages of the chiral nanowires.
Yes. So that is a little bit challenging to think about. So what we do see, at least from what I can talk about right now, is that we see the opening of the band gaps in these materials as we go from the bulk down to the single chain. But in those helical structures, those are quite unique in the way that the interhelix interactions are actually fairly weak. So when we start looking into few chains of that material, the opening of the quantum confinement effect is not pronounced, which actually now the more of an issue, or more of the interesting opportunity to think about is how strain, when these materials would bend and would fold, how those would influence the orbital overlap in the S and P states in the conduction band, whether that will open up or close with strain. So I think that's more of the, in a way, the question that we're trying to look into, because at least for those helices, the quantum confinement effects are not as pronounced. However, in the other analogs, like the antimony sulfides, the bismuth sulfides, and so on, those materials have slightly stronger interchain interactions that we do see quite pronounced of a quantum confinement effect. So we're at the stage wherein we're now actually establishing how much of a quantum confinement effect we observe in these structures by peeling them off or by isolating single chains, because that alone is not known, and that is hard to model with DFT because van der Waals functionals is hard to integrate, and we've seen drastic differences in the calculation and in the measurements. But I think that's something that's, I think, in a way, a degree of freedom that we can start looking into as we assess these materials from the bulk down to the single chain, which that part, the exfoliation of these materials, is not very trivial. We can access single chains in these structures by matching surface tension of different solvent mixtures. But in a way, again, if I'm showing these materials in the context of 4 years with my group working on these, I think the dream as well, the immediate dream, is to get to the stage where 2D materials at, or be inspired by where 2D materials at, that we can now understand many of these, bulk to nanostructure properties and develop ways with which we can even stack these materials and so on. But yeah, thank you very much.
Any other questions or comments?
Everyone had a good lunch, sounds like.
But Max, because when you showed all these banded
Yeah.
structures, so what was the thickness of the...
Yes, very good question. So, so these structures, I'm showing a specific example that's about 100 nanometers thick, but actually we can access this about 20 nanometers thick, at least with our current, sort of this synthesis is not yet fully optimized, but about 20 nanometers thick of a wire, and that is about 500 nanometers wide. So, so something that is quite unexpected and something that we haven't really understood why it happens is that when these materials would grow, we have seen cases with which these would form sort of unmet ends that would twist like a Möbius strip. So those things are still under question, but we're getting into that stage of, as I would assume your question is about, is that in that regime where we might see quantum confinements and confined transport throughout these materials.
So it's still van der Waals is sufficient to keep this single wires together, right?
Somehow, yes. So what happens is we can take these chains and then exfoliate them. So the success actually is still not that high, but it is possible.
Question, so
yes.
you are reading in my mind.
Oh, no, because those are the things that we're interested about, is really trying to see now with 1D materials. It's actually quite interesting in terms of exfoliation, because I work with 2D materials, and exfoliation happens from the ends as well. So it develops strain as we peel these materials off. But in a 1D spaghetti-like bundle, which actually inspired that group photo that we think we're chefs, the solvent and actually intercalants can get into the gap quite easily. So it actually is a lot easier to peel these materials off, even with not that much effort compared to the 2D materials.
And for how long they are stable in the air?
Yeah, my students hate the glove box. So other than that one phase gallium selenium iodide, these structures are stable in air and stable in ambient conditions. So we actually, many of the measurements that we've done in our papers, which I didn't talk about today, are photoluminescence measurements at room temperature at ambient conditions and transport as well. So yeah, it's a now I can't get them to work in the glove box.
So the last question: have you been able, like for example, to look at just a single twisting sequence of atoms, just joining two transistors, or just
yes.
looking at this 1D structure, what happens?
Just
really single, how to say, not a two-dimensional, but one-dimensional.
Just a single chain with few
so
atoms.
The thickness of of angstroms.
Yes, yes. So we can actually, so in one of our papers, we did demonstrate that we can peel off a single chain without the carbon nanotube trick. We haven't figured out the right deposition conditions yet for e-beam lithography, mainly because the e-beam process that the e-beam evaporator that we're using has a strong knock-off energy that we can infiltrate the material. So we're playing this trick of depositing our electrodes in PDMS, which 2D folks have demonstrated, and then stamping those onto our devices, sort of layer by layer assembling the device. So I think that's all thanks to the 2D materials folks that enabled the processing for
Thanks God there's another question. You know, Max, I was supposed to ask questions if other people don't ask people. No, sorry, I'm kidding.
Yeah.
Actually, I have a few. So
yeah, absolutely.
So first of all, so this is the exfoliation method, so like a top-down approach.
Yep.
Do you think it will be possible to build those materials like from bottom up?
Yes, yes.
As a single chain?
Yes, very good question. And actually, that's the reason why I showed points number 2 and number 3 is that was one of the initial goals in my group, is really inspired by work by Ji-Woong Park at UChicago, that they were able to develop monolayer MOS2 in the wafer scale. We're starting to actually, we are asking those fundamental questions of how we can control the nucleation and growth in 1D materials to control just 1D crystallization from bottom up. So there is a paper that we will be submitting this month that shows that we can control the thermodynamic and kinetic parameters well, that we can access not yet single chains, but very, very few layers of a quasi 1D, 2D material from bottom up with wide coverage. So I think we are trying to really redefine how nucleation and growth happens when you have a building block that's already a one-dimensional chain, compared to how back in the days like Charlie Lieber or Pei Dong Yang would work on silicon, which is an isotropic structure with a particle as a catalyst to grow 1D. So now these are exactly the same questions that we're asking of how we can use these chains as 1D building blocks and just arrest the growth. The most success that we've gotten is that example that I showed. So this is from vapor phase, actually. The last example of almost a centimeter long wire. That's about 20 nanometers thick. So we are trying to arrest the growth in earlier times to sort of sacrifice the length for the thickness. But this is one of the most bizarre results that we've found, where a material just continuously grows 1D, and that tells us how we can turn off the interchain interactions. But for now, we were very impatient, and that's why we developed the carbon nanotube chemistry to access these from bottom up single chains that we can look at under electron microscopy as well, as we work on with the other approach.
Yeah, very nice. So, but when you have been showing that you can grow really long chains, right? 245 nanometers.
Oh, no. Yeah, so I'm showing. So this is a single wire from here that goes to here.
Almost.
So that's about 8 millimeters.
Right.
Yeah, almost a centimeter.
This actually answers one of my next questions, because we are living in the carbon era.
Yes. Yeah.
Right. So graphene and so on.
Mm-hmm.
But people are also making one-dimensional materials using graphene, like porphyrin nanoribbons.
Yes.
Or nanographenoids, like long.
Yeah.
So can you compare that to your material?
Yes. So actually, I think if there's any advantage at all, is that when we think about the graphene nanoribbons, for example, there's this zigzag or armchair edge functionalization, which is hard to control. I think for us, when we think about these building blocks are stoichiometrically limited by the phase that it forms. So there's no dangling bonds or uncertainty, and that's how we can model. And this is what inspired this dream, really, is that we can pick a single chain. Current computers can, or our collaborators do not say a word that they can model a single against modeling a single chain. And that single chain is expected to be the same in the structures when we start peeling these off, that there's no dangling bonds that will make these slightly unpredictable, which is critical for these length scales.
Right, this is very nice. But on the other hand, it also limits the branching.
Yeah.
Right?
No, that is the tough question. Is that's the bane and the boon of van der Waals materials, is that they're quite inert. But that's how we started really dialing in on these stereochemically active lone pair interactions that we would find in these structures, because we would get some strength on those interactions beyond just van der Waals interactions. But we're also looking into opportunities. That's why I showed like that indium halogen or gallium halogen bond could be swapped using organolithium reagents with an organic functional group to sort of open up the toolbox. But for us right now, yeah, we're trying to really just understood mostly the chemistry of the inorganic phase.
This is really beautiful and
Thank you.
impressive. Thank you.
Thank you.
Thank you, Max.
Thank you.